A very warm morning to one and all. On behalf of Amity Institute of Pharmacy, Amity University, Noida, and with the blessings of our Honorable Founder President Dr. Ashok K. Chauhan and Visionary Chancellor Sir Dr. Atul K. Chauhan, I, Ramanpreet Walia, along with my teammate Dr. Swati Madan, welcome all of you to the inaugural session of 2 days SERP-sponsored international conference on pharma technology and translational development in chem bio research, traditional to biopharmaceuticals 2024, PTT DCBR 2024, that has been planned to enlighten the delegates from pharma fraternity on recent developments and current trends in pharmaceutical research, like novel drug development approaches, innovations in phytopharmaceuticals, etc. It is planned to focus on encouraging the collaboration and knowledge exchange, bringing the experts from diverse fields, including chemistry, biology, pharmacology, ethnobotany, and traditional medicine, and take up clinical and validation studies for the translation. A rapid progress in molecular research, biotechnology, stem cell research, and genetic engineering led to a paradigm shift in drug development, which is addressing the modern health challenges, including the non communicable diseases, mental health wellness, and to confirm how biotechnology complements the traditional medicines. Thank you to each and every one of you for being here with us today. We also are pleased to welcome the persons who are not able to join us today and they are joining us online. It is such a proud feeling that we have with us a galaxy of intellectuals, benevolent speakers, honorable guests, and wonderful delegates who have joined us from various parts of the country. I once again welcome you all. Without further delay, I invite all the dignitaries on the dais to come forward for the lighting of lamp. Volunteers, please help.
हे शारदे माँ हे शारदे माँ हे शारदे माँ हे शारदे माँ अज्ञानता से हमें तार दे माँ अज्ञानता से हमें तार दे माँ हे शारदे माँ हे शारदे माँ हे शारदे माँ हे शारदे माँ तू स्वर की देवी ये संगीत तुझसे हर शब्द तेरा है हर गीत तुझसे तू स्वर की देवी ये संगीत तुझसे हर शब्द तेरा है हर गीत तुझसे हम हैं अकेले हम हैं अधूरे हम हैं अकेले हम हैं अधूरे तेरी शरण में हमें प्यार दे माँ हे शारदे माँ हे शारदे माँ हे शारदे माँ हे शारदे माँ मुनियों ने समझी है गुणियों ने जानी वेदों की भाषा पुराणों की वाणी मुनियों ने समझी गुणियों ने जानी वेदों की भाषा पुराणों की वाणी हम भी तो समझे हम भी तो जाने हम भी तो समझे हम भी तो जाने विद्या का हमको अधिकार दे माँ हे शारदे माँ हे शारदे माँ हे शारदे माँ हे शारदे माँ तू श्वेत वर्णी कमल पे विराजे हाथों में वीणा मुकुट सर पे साजे तू श्वेत वर्णी कमल पे विराजे हाथों में वीणा मुकुट सर पे साजे मन से हमारे मिटा दे अंधेरे मन से हमारे मिटा दे अंधेरे हमको उजालों का संसार दे माँ हे शारदे माँ, हे शारदे माँ, हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ, अज्ञानता से हमें तार दे माँ। हे शारदे माँ, हे शारदे माँ, हे शारदे माँ, हे शारदे माँ।
Thank you, everyone. Now, let me recite Tulsi Gayatri Mantra. Om Tripuraye Vidmahe Tulsi Patra Dhimahi Tanno Tulsi Prachodayat. Om Tripuraye Vidmahe Tulsi Patra Dhimahi Tanno Tulsi Prachodayat. Om Tripuraye Vidmahe Tulsi Patraye Dhimahi Tanno Tulsi Prachodayat. Om, let me meditate on the goddess of Osimum Tulsi. O goddess who is dear to Vishnu, give me the higher intellect and let Brinda illuminate my mind. On a pious tradition, let us welcome all the dignitaries by offering Tulsi saplings. I now request our Honorable Dr. I now request Dr. Havagire Chitme, sir, Deputy Director AIP, Amity University, Noida, to give Tulsi sapling to our chief guest, Dr. Rajiv Singh Raghuvanshi, Drug Controller General of India. Our VC ma'am will be joining a little late. Now I request Dr. Tanveer Navid, Deputy Dean, Health and Allied Sciences, Amity University, Noida, to come forward and offer Tulsi sapling to our guest of honor, Dr. Arshad Khuru, Head Bioavailability and Bioequivalence Division, Sun Pharma, Gurgaon. Now I request Dr. Vinay Lathar, Professor and Head Pharmaceutical Chemistry Department, Amity Institute of Pharmacy, to offer Tulsi sapling to Dr. A.K. Singh, Senior Vice President, Amity Foundation for Science, Technology and Innovation Alliances, FSTIA. Now I would request our convener, Dr. Havagire R. Chitme, Deputy Director, Amity Institute of Pharmacy, to initiate the inaugural session with a welcome address.
Thank you, ma'am. Good morning, everyone. Godly Founder President Dr. Ashok K. Chavan, sir. Honorable Chancellor Dr. Atul Chavan, sir. Honorable Vice Chancellor Professor Dr. Balvinda Shukla, madam. Esteemed Dr. Selvamurthy, sir, President STIF. Respected Dr. A.K. Singh, sir, Vice President FSTIA. Respected Chief Guest of the function, Dr. Rajiv Singh Raghuvanshi ji, Drug Controller General of India. Guest of honor, Dr. Arshad Khuru from Sun Pharma. Most regarded Professor Dr. B.C. Das, sir, Dean Health and Allied Sciences. Esteemed dignitaries on and off the dais, my dear colleagues, dear students, dignitaries, participants, and sincere volunteers. It is with great pleasure and honor on behalf of the organizing committee of the second PTT CAB 2024 that I extend a warm welcome to you all to this international conference. It is with great satisfaction that we witness this august assembly of distinguished intellectuals, scientists, scholars, and academicians. The conference signifies a noteworthy achievement in our endeavor to integrate the practice of traditional medicine with contemporary evidence-based medicine system. During the forthcoming 2 days, there will be ample opportunity for participants to participate in thought-provoking discourses, share significant insights, and establish substantial network that will undoubtedly contribute to the progression of pharmaceutical technology knowledge by integrating basic and applied science. We would like to extend our sincere appreciation to every esteemed speaker and panelist who has cordially accepted our invitation to impart their knowledge and perspectives. Your invaluable contributions will undoubtedly enhance the quality of our discussions and deliberations. Additionally, I would like to express my sincere gratitude to our partners and sponsors for their generous assistance, which rendered this occasion feasible. Your sustained collaboration is greatly appreciated, and your dedication to our cause is genuinely laudable. We have the ability to effect positive change and leave an enduring mark in our respective disciplines when we work together. I am happy to inform the August gathering that we stressed ourselves to global reach by live streaming of the conference through Zoom and YouTube. I would like to thank our founder president, sir, for his blessings, our honorable chancellor, sir, for inspiring and motivating us, and our honorable vice chancellor, madam, for her wise leadership and guidance. I would also like to thank the president of STIF, the senior vice president of FSTIA, the chief guest, the guest of honor, and the dean of health and allied sciences for being here and addressing this important gathering. I would like to extend a hearty welcome to all dignitaries and attendees again. Together, let us commence this endeavor with excitement, inquisitiveness, and mutual dedication to achieving excellence. Thank you so much, and have a good, sharing, knowledgeable conference for all 2 days. Thank you so much.
Thank you for such wonderful opening remarks, sir. In absentia, with the blessings of Professor Dr. B.C. Das, Dean Health and Allied Sciences Domain, we are continuing with our inaugural session. Now, I request Dr. Navneet Sharma, Organizing Secretary, to give a brief introduction about the conference.
Thank you, ma'am. सभागार में उपस्थित सभी गणमान्य व्यक्तियों तथा देवियों सज्जनों को सुप्रभात और नमस्कार. Ladies and gentlemen, good morning and a warm welcome to the International Conference on Pharma Technology and Translational Development in Chem Biology Research 2024. Hosted by MIT University Uttar Pradesh, sponsored by the Science and Engineering Research Board SERB and Defense Research and Development Organization DRDO. It is an honor to be here with you today as we explore the fascinating field of pharmaceutical technology and translational development in chem biology research in the short PTD CBR 2024. Our theme emphasizes the importance of collaboration across disciplines in addressing the challenges facing the pharmaceutical industry today, from discovering new drugs to translational research into clinical applications. Our discussion over the coming days will lead to groundbreaking discoveries and innovative therapies. PTDD CBR 2024 has drawn over 400 participants from various backgrounds, including pharmacy, biotechnology, life science, and chemistry, showcasing a diverse range of expertise and perspectives around the globe. In the coming 2 days, to examine the scientific basis behind traditional remedies and practices by elucidating the chemical and biological mechanism of action, we are going to cover the scientific tracks of traditional scientific knowledge, stem cells, pharmacology, emergency healthcare, phytochemistry, drug discovery, and drug regulatory compliance. We are going to have the scientific paper presentations of the scholars and students on the different fields of chem bio research. We are grateful to have esteemed speakers from the industry such as Sun Pharma, Mankind Pharma, Abbott, and Emil Pharma, from esteemed research labs of Defense Research and Development Organization, CSIR, IIT, and CCRUM, as well as from UC San Francisco and National University of Science and Technology, Oman. Researchers and students from various research labs of DRDOs, IITs, NIPERs, and university across India and the globe joining us to exchange knowledge, encourage conversations and discussions, and drive forward the field of pharmaceutical sciences. I would like to extend heartfelt thanks to our industrial sponsors Their support has been instrumental in making this conference possible, demonstrating their commitment to advancing scientific research and innovation in pharmaceutical science. On behalf of MT University Noida, I extend my heartfelt gratitude to all the distinguished speakers, delegates, sponsors, and organizations from their invaluable contribution towards interaction, enriching our endeavors, inspire positive changes in the world of pharmaceutical science. Thank you, and let the conference begin. I wish you all the productive and enriching conference ahead. Thank you. Thanks a lot.
Thank you so much, Dr. Navneet. Now, I take this opportunity to invite Dr. W. Selvamurthy Sir, DG, ADSI, and President FSTIA for a welcome address, who is joining us online due to some unavoidable circumstances. Dr. Madan, please brief about Dr. Selvamurthy Sir.
Dr. W. Silvamurthy, President, Amity Foundation for Science, Technology and Innovation Alliances, FSTIA, Director General, Amity Directorate of Science, Innovation, Chancellor, Amity University, Chhattisgarh. His former Distinguished Scientist and Chief Controller, R&D, DRDO, presently member of the Scientific Advisory Committee to Cabinet Government of India. He is Chairman of the Technology Commercialization Committee of ICMR. He is Chairman of Interministerial National Steering Committee of DST on Scientific Social Responsibility and also the Chairman of Assessment Committee set by Ministry of Ayush for accreditation of yoga institutes. In recognition of his significant contributions in the field of biomedical research and development, he has been awarded several prestigious research awards. A few prominent awards are Dr. APJ Abdul Kalam Award for the most prominent scientist and educationist by OSIS World Records 2023, CSIR National Award for SNT Innovations presented by Honorable Prime Minister of India 2012, Lifetime Achievement Award from Honorable President of India in the field of clinical and preventive cardiology 2006. I welcome and request Dr. W. Silvamurthy sir to please address the gathering.
Thank you, Swati. First of all, let me extend a very warm welcome to each and every one of the participants who have assembled here in this very important conference on the pharma technology and translational development in chem biological research, traditional to biopharmaceuticals. Particularly our Honorable Founder President, Dr. Ashok K. Chauhan, who has given a new vision in the education, higher education, and also school education in the country and at the global level. He always says that Amity should make a very significant contribution in transformation of India to Viksit Bharat. He has already, about 20 years ago, he said that developed India, the global superpower as well as general superpower India should become in the next 20-30 years. So such a great person is there today. Founded this Amity group of institutions with 13 universities in India, 18 campuses abroad, 28 schools with 200,000 students, 10,000 faculties, 5,000 PhD scholars. So he has created such a big Amity universe. For what? He wants to create a happy world, healthy world, Vasudeva Kudumbagam, global village, global family. So this is the purpose with which the whole Amity group, education group, he has inculcated this philosophy that we should contribute to the development of 8 billion people. So that is where this conference is coming at a very opportune time when Honorable Prime Minister Shri Narendra Modi ji has given a big vision to the nation, Viksit Bharat, Atmanirbhar Bharat. So all these are not just slogans, but these are the vision which will drive the whole population in India to reach that goal of Viksit Bharat at 2047. So in that direction, how do we, the theme of this conference, deliberations in this conference are very relevant. When you look back, Azadi Ka Amrit Mahotsav, India has emerged as a global pharma hub. If you look at the pharma industries in generics, we are on the top. And also, when you look at the herbal medicine next to China, and we are now catching up at that, the natural products, food supplements, nutraceuticals, as well as the herbal-based medicine and traditional medicines. So India is really emerging as the hub of pharmaceutical, the knowledge, industries, and also economy. So we are very fortunate to have Dr. Rajiv Singh Raghuvanshi. And sir, I wanted to be there personally to greet you, welcome you, but unfortunately due to other commitments, I am not able to be there. But welcome to Amity. We look forward to your blessings to Amity. And we are now, many products are in pipeline, the natural products, herbal medicine. So we would be coming to you for your blessings, guidance, and also advice for taking it forward to the people. And we are also fortunate to have Dr. Arshad Kuru, the head bioavailability and bioequivalence from Sun Pharma, with whom I have interacted right from my DRDO days. The Sun Pharma, DRDO has been a partner with Sun Pharma. Similarly, Amil Pharmaceuticals, we have transferred technology from there on the vitiligo, anti-vitiligo cream, which we have developed herbal based. So I'm so happy to see all my old friends who are here. And thank you, Dr. Arshad Guru, for being a guest of honor at this. And I'm also happy to see our own colleagues, Dr. A.K. Singh, who was the Director General in the life sciences domain of DRDO and who has spent more than 3.5 decades, served the armed forces. So he is now the Senior Vice President driving the APSTIA, Amity Foundation for Science, Technology, Innovation Alliances. And also the other colleagues, Dr. Tanveer Naved, Dr. Navneet Sharma, and all those in the Amity Institute of Pharmacy. You have brought wonderful people, not only in the inaugural, but when I looked at the whole program, everyone, including from academia, researcher, institutions, and industry. This is going to be a very, very fruitful deliberations. We'll be eagerly looking forward to the deliberations. My dear students, please listen to each and every word being spoken of from these experts who have knowledge, expertise, experience, and wisdom. So this is how you are going to shoulder the responsibility of 8 billion people by being the leadership at various levels. So prepare yourself. So students, I would like to listen from each and every one of you what you learned after the deliberations of this conference. And so please be attentive and also listen to every word of it. So India is moving towards $5 trillion economy. Vice Chancellor, Madam, welcome to this inaugural function. Thank you so much. And so India is moving towards that economic power. We are now 5th in the world in economic power. The economy GDP size is about $3.9 trillion. And then we will reach 4 trillion very soon. And then we will add $1 trillion economy every year. So this is going to be our country in terms of economic power. We will soon become the 3rd in the world, for which pharma industry is going to contribute in a significant manner. That is why the theme of this conference is very important, because we have a traditional scientific knowledge as well as modern scientific acumen. So how do we blend these together so that we will be able to be the leader in this field of pharma? That is what we need to make a blueprint out of the deliberations which come out of this. So I'm very happy to see the Controller General of India, who has the whole thing. He looks after the regulatory processes, which he is now has speeded up. The whole process of DGCA operations are phenomenally changed. New paradigm he has given. So Dr. Raghuvanshi, your presence here will also, that address we are looking forward to, will give a new directions that how fast we should move as a country in terms of developing our own indigenous systems of medicine. Because when you look at the pharmacopoeia in the allopathic side, we have almost come to a dead end. How many new molecules are coming? Very few. But then where is the treasure house is, the gold mine is our traditional knowledge. Ayurveda, Siddha, Unani, herbal medicine. This is where we should focus, because India has an advantage of having this biodiversity, 45,000 species of herbal, which has got medicinal applications, which we are not understood yet. We have a hotspot in Northeast, another hotspot in Western Ghats, and biodiversity at the national level with agroclimatic conditions, very conducive for different type of herbal plants and natural products to be harnessed. So this is the advantage India. And we have the root of its practice in India. People are using it almost across the globe. The natural products, herbal medicine is being used. So India has a root of its culture of practice of traditional systems in India. The next thing is there are many institutions and industries have come over a period of last 2-3 decades. If you see the in the Azadi Ka Amrut Mahotsav, when you look back, that we have been able to develop in the last 10-15 years, a large number of the industries, institutions, Ministry of Ayush, separate ministry established, which was a Department of Indian Systems of Medicine, is now a Ministry of Ayush, giving a total focus. So all this is only to bring out, churn out the wealth which is available in the natural products and traditional scientific knowledge. Charak Samhita, if you look at the whole knowledge which has been compiled, that has already laid the pathway to and avenues, opened avenues for many, many new formulations to come from the natural product. But it is for us, the scientists, researchers, institutions, industries, that we should be able to bring this out for the world. Now, the final message I would like to give is Amity is a research and innovation-driven university, and we have a strong pharmaceutical and pharmacological as well as life sciences research strength in Amity. We have a strong Amity Institute of Biotechnology and allied health domains. We are very strong. We have signed MOUs with the many medical institutions, hospitals, including now we are going to sign one with AIIMS, New Delhi. And we have MOU with ICMR, a formal MOU Amity Group has signed with all institutions in Indian Council of Medical Research. So with this, we should be able to give a large number of new products coming. And we have industry. We have a back-four industry which deals with food supplements, nutraceuticals. Amity has an industry which deals with the Ayurvedic products, nutraceuticals, and also products which are being exported to 47 countries. And Mr. Arun Chauhan is now looking after, is the CMD of this. So we have all strength. We have research institutions, we have industry, we have researchers and students population. So with this, our contribution should increase multifold. So my dear students, researchers, let us roll up our sleeves and then get ready to contribute immensely for this transformation, which should happen in the field of pharma. So we are looking now at modern technologies like artificial intelligence. Amity has invested heavily in developing a center of excellence in artificial intelligence with 10 petaflop in the first phase for computing power. And then this is going to be augmented to 16 and 20 over a period of time. We are already going to the second phase. So this has to be used in development, understanding the herbal medicine, the phytochemistry, and also the predicting the pharmacology, identifying the potential active principles, all this using the AI platform. GIS, we have developed a great capability on geoinformatics and remote sensing. Using that capability in Amity, we can do a lot. We have filed more than 2,100 patents. Now we are monetizing the patents, converting them into a product technology process to reach out to the people. So this is Amity's research and innovation. We have published 40,000 research papers in journals of repute. We have reached a H index of 143, and we have also transferred 35 technologies to the industry. So we are already in the right path under the leadership of Dr. Ashok K. Chauhan, the founder president, the chancellor, Dr. Atul Chauhan, now the vice chancellor here, Dr. Balvinder Shukla, and all the members who are driving the research and innovation. Dr. Raghuvanshi ji, we have Dr. Chauhan has created a SNT war room, science and technology war room, in which we have brought about 40 brilliant, young, energetic, dynamic warriors who are driving right from searching a scheme, getting the quality proposal submission, monitoring the sanctioned projects, leading to the outcome and deliverables coming out of the projects, and then also getting into the technology transfer and filing patents. So that is Amity research ecosystem, innovation ecosystem today, which the Honorable Founder President has created. So your presence here is going to be a great motivator for all of us to do much more than what we have been doing. And during the deliberations, many people would speak about some of the outstanding contributions. I'll mention only one. We are now even combining, integrating allopathic and the traditional system together. Dr. B.C. Das, who will be joining soon, that he is the Dean Health Sciences, the molecular medicine and stem cell research. We are using a triple conjugate for cancer treatment. That is a known anti-tumor agent, which is an allopathic, a synthetic molecule, combined with the curcumin, which is from turmeric, and also folate. Folate gives the signature of the cancer stem cell. Curcumin tones down the toxic toxic effects of the chemotherapeutic agent, of the known anti-tumor agent. Then also the curcumin sensitizes the cancer stem cell membrane to take the conjugate inside. So we have gone into the phase 1, and now we will be waiting for the phase 1 preclinical studies, safety and efficacy trials are on. And we have a large number of herbal initiatives. We have established a herbal garden. And in fact, Minister of Ayush, Shripad Nayak ji, came to our campus and inaugurated 15 acres. We have cultivated Ashwagandha, Osimum sanctum, Vitania somnifera, and Sibakthon. So much Sibakthon cultivated here from high altitude. So many such herbal gardens we have established. And so we are in focus both on the synthetic molecules on one side and also on the natural product. So we will be eagerly looking forward to the outcome. We want to work with industry. Sun Pharma is there. Amil Pharmaceutical has joined us. So we are all here. Academia, industry connect is the need of the hour. And that is what Amity's focus is also. So I welcome each and every one of you for this wonderful, very important international conference. We will be looking forward to the outcome in this chem biology research using traditional and biopharmaceuticals field. And best wishes. And finally, I'll conclude with Dr. Chauhan's words that outcome only matters, because we spend a lot of time, efforts, money. Each one of our time is precious. So we should have a fruitful outcome from the deliberations. We will be eagerly looking forward to the major recommendations from this blueprint. What should be the focus, priority areas of research and development, so that ultimately it will contribute to Viksit Bharat. Best wishes to each and every one of you, and thank you so much for joining us.
Thank you so much, sir. It is an inspiration always. We are blessed to have you as our mentor and guide. You are not only a distinguished scientist, but diverse in technology as well as in the traditional system of medicine. You are such ocean full of knowledge, sir. Thank you so much. Now, as our Professor Dr. Balvinder Shukla, ma'am, our Honorable Vice Chancellor has arrived. So I would request Dr. Tanveer Navid, Deputy Dean, Health and Allied Sciences, Amity University, to come forward and offer Tulsi sapling to our Honorable Vice Chancellor, ma'am. Thank you, sir. Now I request Professor Dr. Balvinder Shukla, ma'am, Amity University, Noida, to kindly address the gathering with your words of wisdom. No words can express your ocean full of knowledge in multidisciplines of education. Dr. Madan, I would request you to come forward and introduce about our Dr. Balvinder Shukla, ma'am.
Most respected Honorable Founder President, sir, Honorable Chancellor, our Honorable Chief Guest, Dr. Rajiv Singh Raghuvanshi, Drug Controller General of India, our esteemed guest of honor, Dr. Ashud Puru, Head Bioavailability and the Bioequivalence Division, Sun Pharma, Gurgaon, my esteemed colleagues, Dr. Silvamurthy, Dr. Das, Dr. A.K. Singh, Dr. Naved, Dr. Chitme, all the faculty members, and dear students. First of all, I, on behalf of Honorable Founder President, Honorable Chancellor, Deans, and Head of Institution, extend very hearty and a warm welcome to our distinguished and honorable chief guest, guest of honor, and all my colleagues for the International Conference on Pharma Technology and Translational Development in Chem Biological Research, traditional to the Biopharmaceutics 2024. I would like to congratulate the team of AIP for putting in this conference in place. And Dr. Silvamurthy has given very detailed introductory address of what Amity University is doing in this sphere, because the vision of our Honorable Founder President is to contribute to the nation building through research, education, and beyond. And there is a huge focus on research and innovation in this university, where even the research component is integrated in the undergraduate programs, in addition to the master's program and the doctorate program are the research focus anyway. This conference is at a very right time. One side, the vision of Viksit Bharat, where with the start of the Amrit Kal, by completing the 75 years of independence, our Honorable Prime Minister has given the roadmap and the dream of India that by 2047, the India to be the fully developed nation. Not only that, but has also given the a vision with the 5 sectors, starting with the empowered citizen, and the last one, that India being the Vishwa Bandhu, and where the in between is the technology, innovation, industry, and economic sustainability. All those aspects are there. And when I was going through that vision document, and the first time seeing that how that vision document is for all, and different stakeholders have to work and act on it to achieve the ultimate goal. So it is the same vision document for the industry, what it is for the academic institution, and the message and the common line is that how the industry and academia can work together. Whether it is for education, whether it's for skill development, whether it is for the research, innovation, translational research, entrepreneurship, which all ultimately contribute to the economic development and India becoming the fully developed nation. And our Honorable Founder President Dr. Ashok Chauhan, way back in 1997, when in one of the addresses, when he was giving his visionary thought, and he said that my dream is, and my passion is, and my junoon is India to be the superpower by 2030. And since then, he has been relentlessly working for this one goal of making India a superpower. And his major focus is that how the student be the success story on one side and skill developed, value-based education, but simultaneously focusing on research, innovation, and the technology transfer one side from that innovation and the other side, starting the ventures. And when we were looking at this vision document, And I really looked at how the Honorable Founder President envisioned long time back. And the both vision document and what the Founder President's vision was, that there is so much the similarity or alignment. And we're really thankful to our Honorable Founder President. And this vision document is talking that India becoming the Vishwa Bandhu. Our Honorable Chancellor always believed in the youth power. And he said that the youth of Amity, youth of this nation, with value-based education, will not only think about themselves, but they will think for the nation, they will think for the society, and they will think for the society across the globe and bring those changes which will make this world a better place to live. Because ultimately we are one world family, and G20 has emphasized amply that the one earth, the one word, the one family. And how can we contribute to that goal for the Viksit Bharat? And this conference, and I'm so happy and delighted that Dr. Arshad Kuru, the head of bioavailability and the bioequivalence division of some Pharma, Gurgaon, is here with us. Thank you very much. And also the Honorable Chief Guest, Dr. Rajiv Singh Raghuvanshi ji, Drug Controller General of India. And we would seek your guidance that how the pharma sector, what all the initiative in light of the Viksit Bharat vision document, what all we need to together, and what Amity can do for the industry, and how what industry can do with the academic institution, that not only we are developing the future ready skilled manpower, but also leveraging on the research innovation and the academic institution working on the industry problems. Finalizing what will be the in next 5 years, then the next 5 years, and we make a roadmap up to the 2047. But the student who take admission, say this year, 4 years down the line, they are going to pass out. What kind of the skills that will be needed 1 year down the line, 2 years down the line, 3 years and 5 years? And that has to translate back to the curriculum, the mandatory courses, the core courses, and the value addition courses. How the way the industry can add that value that the industry gets at the end of the day, what they are really looking in the terms of skills, knowledge, and attitude, so that that talent pool starts contributing to the industry from the day one. And we would like to discuss and deliberate for this, so that when we start the next academic session 2024-25, we are ready with that kind of the curriculum, that kind of the pedagogy, that kind of the value addition that we always endeavored. And it's the Amity, the first university in the country which had the corporate source center from the day one when the higher education institution was established. And Honorable Founder President at that time envisioned, he said, no education is complete without having the connect with the industry. So many things, the value-based education, Honorable Founder President said, it's very important today when we are looking at the Viksit Bharat or National Education Policy 2020. Everything, we find that, so much has been, what already our Honorable Founder President has emphasized in the education. And with this, I would like to once again welcome our the guests. I'm very thankful to, I request all of you to please extend very warm welcome to our Honorable Founder President. Thank you very much, Honorable Founder President. And we know that your passion, your interests, everything lies that how we connect with the industry and develop the industry ready talent one side, and the other side that how to engage with the industry for research, innovation, translation research, and the entrepreneurship development. Thank you very much for that vision. And when we are looking at the vision document of India, of Viksit Bharat, and and your vision right from the beginning when you started a school education, a higher education, and we are saying that it is your vision which is echoing with the now the which is the honorable Prime Minister has given. So your dream of making India superpower and the Viksit Bharat, that will be realized. And we are having the industry partners here. And I think that the kind of the industry partnership Amity has, Amity is being the largest training provider in the country, skill based, capacity building based, the training program, but our dreams are much bigger than what we are doing at the moment. And we want to be the world's number one training provider, the skill development, capability, capacity building program, because that only will make India when it comes to the Viksit Bharat. Thank you very much, Honorable Founder President, for having the present in this conference. Thank you very much.
Great.
Thank you so much, ma'am. It is a great pleasure listening to you again and again. With your leadership, we are continuously improving in our abilities to perform better every day. Thank you once again for your enlightening words. We are really honored, ma'am. Now, I would request Professor Vinay Lathar, Head Pharmaceutical Chemistry Department, Amity Institute of Pharmacy, to introduce our guest of honor, Dr. Arshad Khuru, Head Bioavailability and Bioequivalence Division, Sun Pharma, Gurgaon.
Respected Honorable Founder President, respected dignitaries on the dais, dear colleagues, faculty participants, and delegates from different organizations, good morning all. It's my pleasure to have you all here today on the occasion of the inaugural moments of two-day international conference on pharma technology and translational development in chem biology research, traditional to biopharmaceuticals. Today, we are honored to have with us Dr. Arshad Khuru, a renowned scientist with over 3 decades of experience as guest of honor. He graduated in pharmaceutical sciences from Jamia Hamdard, did his postgraduate in pharmacy operations, and accomplished his PhD from BITS Ranchi. He started his career as a bioanalyst in Ranbaxy Research Labs and currently is heading the bioavailability and bioequivalence functions at Sun Pharmaceuticals India, Gurugram. Dr. Arshad has 28 years of extensive experience in conducting bioavailability and bioequivalence studies for domestic and international markets. He has been instrumental in developing GCP, GLP processes at Sun Pharma. He has been honored with several awards like Team Excellence Awards from Ranbaxy and Hakim Abdul Hamid Award from Jamia Hamdard. He has authored more than 100 publications in international journals of repute. He is passionate about management development programs. He has conducted more than 35 workshops in universities, NGOs, and overseas corporates on topics like collaborations, effective communication skills, and self-motivation. He has authored books entitled Echoes of Experience and 30 Insights from Life's Journey. Once again, we are grateful to have Dr. Khuru for joining us today and share his insights. I request Dr. Khuru to address our delegates.
Morning, everyone. Thank you so much. Actually, it's my honor to be here with our distinguished chief guest, with the distinguished VC, faculty members, students. I will not take much time because I have one talk after this inauguration session is over. I have one talk with you wherein I will be presenting some talk on bioanalysis, that's evolution in bioanalysis. But I want to just spend a few moments here telling you that what's the importance of these conferences or workshops. This international workshop has at least 3 benefits. There are multiple benefits, in my opinion. I can give a talk on the benefits of the workshop of this conference, but 3 benefits I want to tell you. One important benefit is that you get a chance to present yourself, to present your innovations, to present your discussions, to present your thoughts in front of many participants. Second important benefit, which we all know, as a participant, you get benefit to listen to those talks, to listen to those inventions, to listen to those functions. But third important thing, which I think is being ignored, which I want to highlight here, is that this gives an opportunity for building a gap or building a bridge between industry and the academics. Through these conferences, we industry people, we actually come to know mainly about academics, what's happening in academics. When I came here in this campus, unfortunately, for the first time I came here, it was unfortunate I couldn't see this campus before. I was amazed to see a lot of students here. I was amazed to see a lot of people here, a lot of faculty, and she told me that there are 48 students in MIT University in this campus only, and I was amazed. That's why I told you this third benefit is the most important benefit, wherein we can develop that bridge between industry and academics. I look forward discussing with you on different topics, on different discussions, and I will definitely see all of you at around 12:15 in my talk. And thank you very much for that. I will not take much time of yours right now. Thank you.
Thank you so much for your captivating words, sir. It had really charged everyone to gain knowledge further. Heartful, warm words of welcome to our Honorable Founder, President Dr. Ashok K. Chauhan, sir. We are waiting to listen to your motivational words and seek your blessings full of ebullience. Sir, you always inspire us to do more and more and have always supported us in all domains of professional as well as personal lives. Words are really less to speak about him, but to sum up his mission is nation building through education and beyond. A big round of applause for our founder, President, sir.
Great. My dear students, your institution is very close to my heart. Many, many years back, I used to come quite often because there is engineering, there is biotechnology, there are other topics, artificial intelligence, but the pharma is the most important. If anybody gets sick, then we from pharma will treat them with the medicines, the right treatment. So therefore, I would say that all students, research scholars, fellows, those who are in this institution, I think they are very lucky to have opted for this branch for their studies. The world is taking a great change. जो हिंदुस्तान की तरक्की हो रही है, I think we all know. इस समय गुजर रहा है, ऐसा ना कभी पहले आया, ना कभी आएगा. So it is for us golden opportunity to achieve all our dreams. Whatever we dream, the atmosphere is there, the environment is there, the possibilities are there. Therefore, to my most beloved students, I will say, have a high mission in your life. Today, whatever mission you will set, you are bound to achieve it. And if you do not achieve what you have dreamt, then it is my mistake of my deputy director, Naved, and all others. It means we have not trained you, taught you properly so that you could not fulfill your dream. I will soon come also to your institution, but I'm very happy to see that this program has been made in such a way that it will be very important for you. Just now also, our guest of honor gave very good tips within few minutes. And I must tell you, I have read about Dr. Rajiv Singh Raghuvanshi ji. He is great person in the pharmacy. To be drug controller is very, very difficult thing. There are so many complicated things in the country, linkage with overseas. So I would say, Dr. Raghuvanshi ji, we are so proud and so glad that you could spare your time. Actually, I have to rush now, but I told I must greet Raghuvanshi ji. I must greet Furoji, the guest of honor, and also my people, those who have introduced Mr. Hargire Singh Chille. अभी ये नया है तो इनका नाम मुझे समझने में बोलने में टाइम लगेगा. But I welcome you. You have joined us. Very, very wise decision. Anybody who joins MIT, he will have a joy that MIT is going to be #1 university in all fields in the country. Yesterday, I dictated a letter, make a 5 years white paper that our university in 5 years is #1 in the country and one of the best in the world. Why should it not be? When drug controller Raghuvanshi ji इतने busy होते हुए आए हैं, it means they know that when we go there, spend our time, it is worth motivating the students. And I would say that I will come soon, but my dear student, have a big aim. If you cannot achieve in MIT whatever you dream for, I heard Dr. Murthy ji also, I had Vice Chancellor, but unfortunately I have to rush for a meeting 12:30. In election days, one has to rush here and there. So to all my guests, my heartiest thanks, my gratitude, and I am obliged that you have come. My dear student, I will come once again. I am very happy that such a big number you have attentive, because when I appear somewhere, I told to my IT people, I want to see the faces of my student, my other people sitting in the hall, because that tells me the pulse of the student. That tells me the power and synergy and energy of the students. So I must compliment all the leaders of the institution, that our students are so good that with them we can have the joy that your institution will be number one. Your former director wanted to go to Mohali, and our chancellor has transferred him to Mohali. He was also a great deliverer, but I always see the comfort of my faculty, my directors, if they want to go with family there. So I agreed that he should go there. But I think our new in charge, Dr. Havgiri, is a great person, and he will bring to the greater size. Once again, our drug controller साहब, मैं आपका बहुत बहुत आभारी हूँ. आप आए, मैं आपसे मिलूंगा कभी. I will find out where you are sitting. आप सर दिल्ली में बैठते हैं, कहाँ बैठते हैं? I'll find out. पर मैं आपसे मिलूंगा, क्योंकि ड्रग कंट्रोलर इतना मुश्किल काम है. मुझे बिठाए तो मैं 24 घंटे में मिल्खा सिंह की रेस से भाग जाऊं. मैं नहीं बैठ सकता आपकी कुर्सी पर. But I'm grateful, and my blessings to all of you. And now I take leave. Dr. Murthy, you are attending also Manas Mandal's lecture also?
Yes, sir. I just attended and came back, sir.
Right. Thank you very much. Thank you very much.
Thank you, Founder President. Your words of wisdom is always a great inspiration for all of us. It's a blessing for all of us.
Dr. Modi, I take the inspirations from my students, faculty members, research scholars, and the institutional heads. Yes. Because inspiration is also taken from somewhere. They are so motivating, so inspiring. And today I'm dictating you the white paper for 5 years. In 5 years, I want to win S&T, research and technology and STEM, #1 in the country.
We will be.
All organizations, government or private, my scientists are so unbelievably good. Students are unbelievably good. Why should I not be #1 in 5 years? Thank you very much.
We will be, sir. And you have given a word that it will be realized. What you dream happens. So it is happen.
Great. Thank you, sir.
Thank you so much, sir. We are really blessed to listen you always. Now, it is a matter of immense pleasure that Dr. Rajiv Singh Raghuvanshi, Drug Controller General of India, has been bestowed with honorary professorship at Amity Institute of Pharmacy. A big round of applause.
Dr. Raghuvanshi Saab, grateful and congratulations and grateful for accepting this honor from Amity University. We are blessed by your gracious presence and gracious acceptance of this honorary professorship. Now you are part of us and we are part of you.
Now I would request now I like to call upon Dais Dr. A.K. Singh, Senior Vice President, Amity Foundation for Science, Technology and Innovation Alliances, FSTIA, to introduce our chief guest, Dr. Rajiv Singh Raghuvanshi, Drug Controller General of India.
Namaskar and good morning to all of you. I think it's good afternoon now. Honorable Founder President, Honorable Vice Chancellor, Honorable Chancellor, Dr. Silvamurthy, who is online, and Dr. Rajiv Raghuvanshi, Drug Controller General of India, and also today's chief guest, and Dr. Khuru from Sun Pharma, Naved, and HOI of Pharma, Barge, I'm also learning to pronounce, then Dr. Lathar and Dr. Navneet, and all senior faculty members, delegates. I have come to know that 400 delegates are registered for this conference. And I also have one of my old colleague from DRDO, Dr. Hemant Pandey, who is also sitting in the audience, and Dr. Murthy and other people. I think before I do the job of introducing Dr. Rajiv Raghuvanshi, I'll take few minutes, just 1 or 2 minutes, to share my thoughts on this conference. I think nothing could be better title or topic of this conference in today's context. One thing which we have seen in COVID, because I come from DRDO, people used to say that the weapon lobby is the mightiest lobby in the world. But during COVID, the pharma industry outshined the weapon lobby, and today the pharma industry is the mightiest lobby or mightiest industry in the world. And today's audience, this cohort, is part of that industry. That is the great thing. And the another shift which we are witnessing and we witnessed during COVID is the change from chemicals to biologicals. Though it had been taking place, but the speed has enhanced during COVID and post-COVID because there were so many lessons which we learned. Let's not go into the details. I'll say now the shift is from generic chemicals to specific biologicals, or I'll say the addressed envelope biologicals. That kind of shift we are into, and the whole onus and challenge and responsibility is on this cohort, that how do we make this challenge or convert this challenge into the opportunity. I think few days back I was reading one article, a report of Deloitte, that what would be the status of biopharmaceutical in 2047. I think he predicts that 5 shifts. I'll just name them. He says that things will shift from preventive to early detection. I think we all know. Then customized treatments. Customized. Then curative therapies. I think that is one major aspect which is going to drive. Curative, because it has got, its canvas is so big. And we are witnessing 1 or 2 things. I think Dr. Rajiv Raghuvanshi may be part of this. I was reading the CAR T cell therapy, I think, which we are witnessing in India now. Then digital therapies and precision interventions. When I say precision interventions, that machine or digitization of equipments for treatment and therapy or surgery. I think we had a very wonderful lecture by Dr. Kale, who really took us through that how the digitization or automation is bringing precision in diagnosis and surgery of tumors. That's all. Coming back to my main topic, I think today is a great day. I think great day for MIT because we are fortunate to have amidst us Dr. Rajiv Raghuvanshi, who is Drug Controller General of India, the chief regulator, which is a very responsible, sensitive position where he has to be very careful in approving the drugs, analyzing the clinical trials, quality, and pharmaceutical standards. And I think he is doing that job very, very meticulously. And, before that, people didn't know about who is DCGI, but during COVID, it became a household name. हर आदमी को पता था कोई regulator है, अगर वो दवाई वैक्सीन को clearance दे देगा तो हमें दवाई वो वैक्सीन मिल जाएगा. तो राजीव, your designation became a household name during COVID. And Rajiv, what I find, because he happens to be my friend, long known. I think I know him for last about 35 years. I came to know him through one of our common friend or common senior, Dr. V.S. Chauhan. And I had been seeing him since then. He's a unique amalgamation of knowledge, manager, leader, administrator, and over the above, a problem solver. And I think that fits very well into his profile of today, or the responsibility of today. And the position of DCGI has always been occupied people with government background or some seniors from the PSUs. But with Rajiv, it is the first time that DCGI, who comes from the corporate background, because he had served in multinational, pharma multinational. If we go to his journey, he is alumnus of IIT BHU. He did his bachelor's, master's from BHU. Then he did his doctorate from National Institute of Immunology, a very high institute with high credibility. And where his main interest in PhD was development of extended release vaccines by which he could reduce the dosages or number of injections. Then after PhD, he preferred to go to the industry. Then he served in Ranbaxy, where he was working in the development and registration area and launch of NDDS, generic and branded generic in global market. He stayed there, but contributed for 12 years. Then again, he shifted to another multinational, Dr. Reddy's laboratory, where he served for 11 years. And there he got 6 products approved by first review cycle of US FDA. And during his tenure in Dr. Reddy's lab, he got the opportunity to interact with US FDA and the regulators of other countries like South Korea, Sweden, Romania, etc., etc. In Reddy's lab, he also served in the production innovation division, where he expertised in the dosage form, particularly in the solid dosage, means oral dosage, either solid, oral, or powder, etc., long list, nasal sprays, extended release, sustained release, etc. And to his credit, he's a great researcher, an avid researcher. He has 14 US granted patents and 250 published PCTs and Indian patents, 25 publications in peer-reviewed journals, and he has co-authored 6 book chapters. Beyond this, he is also a visiting faculty to NIPER Hyderabad, NIPER Mohali, and IIT BHU. And though he is a regular, you can see him in various conferences, national, international, various meetings of pharmacy, where you will find him either inaugurating or as the chief guest or a resource person. He's a scientist, he's a person who got the Dr. Reddy's Excellence Award not once, twice. When I said he's a leader, many of his subordinates are at the, leading positions in various pharma companies. He's a leader who has also developed so many leaders. Then I think after about 23 years, he decided to come back to government. And that is the time when he joined Indian Pharmacopoeia Commission. And that was also the time when I again came in his contact in the capacity that I am a member of their scientific advisory board, and I meet him. And let me tell you, he has brought so many changes in IPC, which I have seen while attending meetings. And I find that the amalgamation of, the corporate experience and then translating into the government setup is really bearing fruits. He brought many changes, like he enhanced the, number of impurity standards, usage of Indian pharmacopoeia, and digitization of Indian pharmacopoeia. Pharmacovigilance and materiovigilance, he gives a special emphasis. And another thing, he's a man who predicts. He created a committee to look into the biological impurities. Though it may not be a concern of today, but tomorrow it is going to be a big concern. And he introduced it right now. Now, simultaneously, then it was 23 February 23. 23 February 23 को he took over as the chief regulator as DCGI of this country, which it's a great responsibility. And, I'm lucky to be associated with you, Rajiv. You are a good human being. And as DCGI, he has brought again new changes. He got IP recognized in many foreign countries, Afghanistan, Ghana, Nicaragua, etc. And he also enhanced the number of IP standards to 678 and impurity standards to 552. You may not appreciate the numbers, but that is the third highest in the world. Became member of the Pharmacopoeia Discussion Group, Europe, Japan, and US, our IP. And he also created risk-based inspections of the pharma company to take care of the quality practice and good manufacturing practices. Rajiv, you are a great person. We are so lucky to have you with us today. And on behalf of Founder President, I welcome you again. Thank you.
Good afternoon. Good afternoon. Thank you. Respected Dr. Ashok Chauhan ji, Dr. Selva Murthy, my dear friend Dr. A.K. Singh, dignitaries sitting on the dais, friends, delegates. It's a pleasure to be here this afternoon. But before I start, I would just like to spend a couple of minutes on why did I repeat the good afternoon. So there are two very peculiar characteristics of Indian society. One is we need to be pushed to do something. We don't do it in the first go, which ultimately goes back to the psychology that we hesitate in taking initiative. That is one. Second is, you will invariably find the front rows in a gathering always vacant. And people need to be pushed to come and sit on the front row. that's also a characteristics of Indian society. It's because we have inhibition in leading things. Historically, we have been followers. Unfortunately, history of this country is either we were ruled by the kings and thereafter we were ruled by British regime for long. So psychology actually has become to follow and not to lead. And it has given us a psychology which creates self-doubt that am I so good? If you think in and introspect, you will invariably see that this question comes in your mind that actually I am so good. Am I so good? And that brings us to a situation where we avoid facing something, we avoid being in limelight, and ultimately we avoid to be leaders. And that is probably one of the bigger weaknesses which as a country today, the country's leadership is addressing. And you have seen the change in last 10, 15 years that we started from where and today where we are. So it's a matter of realizing that we have that potential, that if we कुछ ठान लेते हैं तो करके मानते हैं. And the latest example is the Chandrayaan success for this country. And you will see many more to come. So, as I said, very happy and great to be here in this campus. And as my old friend Arshad was saying, I have also unfortunately come to this campus for the first time. And since my education is from government setup, where size is what you see, probably one of the biggest campuses in this country from where I come. But the kind of infrastructure which I see here and the campus which I see here, I have traveled throughout the world and I see anything and everything is comparable to anything which is of world class. So a infrastructure which your founder has given, and the request as he was also making and everybody would have, that don't let it go waste. A lot of blood and thinking has gone in this, and the onus is on people sitting here, all the students, the future of this country, to make use of that to the best possible extent and give it back to the institution and the society. So why I always like to be in this kind of atmosphere which is there in MIT, because of mainly two reasons. One is the young crowd. We see a positive energy all around, and that is very difficult for people like us to feel in daily life. And the second is because this is a place which is a center of creating knowledge. And when I say center of creating knowledge, it has so big an importance in a country like India in the present context, that the future of this country lies in hands of people sitting in this auditorium, people who are young and who are actually citizens of tomorrow. When you go out of this campus and start contributing to the development of society, that creates the future of this country. So a lot lies on what kind of knowledge is being created in these campuses and how those knowledges are being imbibed by people who are staying in these campuses and carry those knowledge when they move out and become part of the working society and get an opportunity to pay back. So that is the importance of these kind of campuses, and that also brings me again and again, whenever I get opportunity, to come and interact with this kind of audience. So I can speak for hours, but unfortunately, today may not be the time, but I would love to come back again. And now, as this university has been kind enough to give me honorary professorship, so it becomes a responsibility for me, and I would wait for the first possible lecture which I can actually deliver for students and teachers and the community, where I can share my experiences of last some 30 years, right from my student life to date. And you will see it's an example of how we can actually develop ourselves, which ultimately is not development of the individual, but ultimately it's development of the society. But before that, for today, just to bring you to the context. So as told during introduction, I'm holding the head of National Regulatory Agency position today, and heading regulatory system for world's most important country from pharmaceutical point of view today is challenging, is interesting, is it satisfying, but it's also very, I won't say stressful, because stress is something which depends on individual, but it's sensitive, because whatever decision we take today, it doesn't impact only India, it impacts the whole world, because India has been designated, and rightly so, as pharmacy of the world. You will be happy to know, some of you must be knowing, that we are doing a pharmaceutical trade of 50 billion US dollars distributed between domestic and export, 50-50, 25 billion domestic trade, 25 billion exports. We export medicines to more than 200 countries in the world. We produce 60% of global vaccine production. We send more than 25% of total medicine consumed in England, United Kingdom. We produce and send more than 40% of generic medicines consumed in US. So all this is responsible for giving us this designation of pharmacy of the world. Reaching on top is very difficult, but sustaining the position at the top is more difficult. And there comes the role of people like you to keep that sustainable, that designation, that position, and more than anything, the opportunity to serve the global population, the humanity of the world. We need to be sustainable in pharmaceutical domain so that this position keeps on growing. Because in management jargon, anything which does not grow dies on its death, dies its own death. अगर जिंदा रहना है तो ग्रो करते रहना पड़ेगा, अगर स्टेबल हो गए एक लेवल पे तो मर जाएंगे किसी दिन. So the ambition for Indian pharma industry today as a country is to reach to a trade value of 130 billion US dollars by 2030, next 6-7 years. Today we are around 50. So we have to actually grow this whole trade by almost 3 times, 2.5 times. This will not happen through our generic model of business which we are doing today. We are third largest producers of medicine by volume in the world, but we are 14th largest in terms of value. And calculation is simple. It's not something very big science. Because we are in generic business, we produce a lot of volumes, but realization in terms of value is not proportional, because cost of generic, the whole generic model survives on low cost. That is why the realization in terms of value is not as big as the volume which you produce. So government today is bringing out all the policies to support the shift from volume to value. And volume to value shift will happen only if there is innovation in the system. The products which we produce have to be innovative, and we'll have to shift from the mindset of generic medicine production to innovative medicine production. And for innovative medicine production, there has to be training in these kind of campuses which produce brains which think innovative. As I said in the beginning, we are very, because of whatever historically has happened with us, we are very good, probably best followers in the world. If we are told to do something, we would probably do best in the world. But we wait for somebody to tell us that please do this. It can be extended to the generic success of generic model of business in this country in pharma is also a reflection of same psychology, because somebody gave us a reference material and told us to copy that material. And we were quick enough, we were innovative enough, and we did it so good that we became best in the world in developing generic product. But that we could do only because we had something to copy. The moment it comes to make a product which has to be copied by others, we start faltering. Because we do not have that original thinking. We wait for people to tell us, guide us, and then once we have that clarity to what to do, then we start running. But this is the time when that need to shift, that mindset need to change. Don't wait for people to tell that what to be done, but you prepare yourself to guide people that what has to be done. And then we will become innovators, and then we will be hitting that $130 billion aspiration by 2030 and become world leader in pharmaceutical based on the innovation, not on generic business. And here comes the role of institutions like MIT, where you have to create that atmosphere, which we call in management terms an environment of curiosity. If you see the teachers, the leaders of these campuses, they have to create that atmosphere of curiosity among students who start asking questions at every point. Today we feel inhibited in asking questions also. And I can bet if I tell you from this audience, stand up and start asking question, you will wait for first person to start asking question. And the moment somebody stands and asks first questions, then everybody will start asking question, and we'll have to then cut it short because of time. But the first question, it will take time. Unless until you will be pushed to ask question, you will not ask questions. So that is because we do not have that culture of curiosity also built in the system. So request to people sitting in the front row, the teachers, the leaders who are shaping the future and the mandate and what has to be done for the campus and the students' curriculum, please develop that culture of curiosity, which will ultimately bring the thinking processes within ourselves, within the students, and then you will see that miracle, that shift will happen where people will start developing things which are for the first time in the world. If you scan this whole country's startup domain today, you will not find more than one or two, which you can find, count on your fingers, startups in pharma domain. 99% of startups today are in either technology or service industry, and everything is backed by technology. And unfortunately, you will not find many of them. At least I have not seen any successful startup in pharma domain. And that also reflects on our confidence, our risk-taking capability, and of course, our capability to ask questions and then start finding answer for that. So it's a big gap. So students need to be focused. And on the lighter side, I see a lot of focus today in this audience also, especially second, third rows behind. But that focus is on their smartphones. 25% of people are on their phone, not listening to what I am speaking or what my predecessor spoke. Use social media, but use social media for bringing those positive energies, positive networks, which helps you in building your career and ultimately contributing to building the society for this country. So we need thinkers, we need these universities and these campuses to produce thinkers and not followers. And I think I'm amply clear that why do we need that? So I said one of the qualities which are expected from successful, innovative people is their capability to take risk. I remember when I was in my final year of post-graduation, we actually used to get some scholarships also while doing M.Pharm. So my dad's first advice to me was कि बेटे अगर कुछ पैसे बचते हों तो उसके FD करा लेना. That was the psychology of a defensive culture and a protective father कि जो पैसे हो उसको बचा लो, वो सिक्योरिटी में डालो कि तुम्हारे फ्यूचर में काम आएगा. Because this society was completely risk averse. Capability नहीं थी risk लेने की. Because we had a past which never supported the culture of risk taking. Ab to, I mean, the times have changed, and you guys have to take advantage of this context, which is a golden age for this country, where you can take risk. And very interestingly, Vivekananda's quote is so apt in this situation. Vivekananda has said that it's the easiest thing to take risk, because if you take risk, if you are successful, you become leader. If you fail, you become guide. So there is no failure in taking risk. So jump into it, start taking that risk, because this generation has nothing to lose. Sometimes I feel scared when I see my daughter. For lots of them, there is no motivation because they have everything available on their platter. हम लोग तो मतलब struggle करते थे इसलिए कि we have to grow in our life, establish, grow, excel. Very interestingly, when I joined my first year B.Pharm, in the first day of ragging at IIT-BHU, I was asked what is the spelling of pharmacopoeia. And as luck would be, I didn't know what was the spelling of pharmacopoeia. And I never dreamt even that I'll become head of pharmacopoeia in this country. So what is required is an honest effort, an ethical individual, sincerity, perseverance. For rest of the things, there is somebody who plans everything for you. We have to do what is in our hand, and that is a sincere effort with ethics, honesty, and sincerity. And things will come, whatever you want will come into your journey. So before I end, just two things which I normally quote in this kind of gathering, which I think would be very helpful to all of you when you move out of colleges. A lot of you must be having your network, your seniors working in industry, or you may be reading in press, industry में बहुत stress है, काम करना बहुत difficult है, huge workload. But fact is that stress, whatever we define, stress is essential in the system. Unless until there is a stress, you will not actually bring out the best in you. What is not good is strain. But stress is always good for you. We mix up stress with strain. We should avoid straining ourselves, but stressing बहुत जरूरी है. There are two very apt examples from nature that why stress is important. First example is of butterfly. If anybody has read, or people like me who had hobby of basically breeding butterflies. When I was kid, I was in hostel from class 1 to, throughout my life I studied in hostel. So we used to go to jungle and bring caterpillars. एक शू बॉक्स में थोड़ा होल करके उसमें डाल देते थे मदार के पत्तों के साथ. 2-4-5 दिन में प्यूपा बन जाता था, उसमें से बटरफ्लाई निकलती थी. We used to enjoy. But later on, when you basically reflect what you used to observe, it is a known fact and scientifically proven that if you help butterfly come out of pupa by dissecting it or breaking the pupa, the butterfly will never be able to fly. It will die its own death. Within minutes, you will see butterfly trying to fly, but she cannot, and ultimately she will die. Butterfly has to come on its own out of pupa with its own efforts, and the moment they start flipping their wings, they get the strength in the wings, and then only the wings will be functional and they can fly. So they need that effort which puts stress on the system, which gives the strength to the wings, and then flying happens. Second very interesting example. I don't know whether you have observed that or not. Bougainvillea, those creepers, very beautiful color. Sometimes you will see the whole creeper is actually full of red flowers. You will not see a single green leaf there. And sometimes you will see it is only green leaves, no colored flowers. And there is a reason for that. It's not it's one variety or the other variety. There is a reason for that. If you water bougainvillea on daily basis, you will get only green leaves. If you want to get colored flowers in bougainvillea, you will have to basically starve them of water. Don't give them water. Then only, because of the stress in the system, the new leaves which come, they are actually colored. For us, it's nice to see, but it has come, when you see that plant or that creeper, that has come through stress on the system. आप करके देखना. This is we used to think that कि मतलब किसी के घर में गमलों में इतने अच्छे बोगनविलिया कैसे होते हैं, और हम लोग because of love to the plants, we used to water on daily basis. Daily evening उसमें पानी डाल दो, and वो कभी भी colored flower नहीं आते. Somebody smart told us that try this. And today, if you come to my balcony, you will see various colors of bougainvillea because we have restricted water to that plant. So, develop that habit of stressing yourself to help yourself to bring out that best in you, which would actually help the whole society and the whole world to grow. And this opportunity of being in pharma world, in the anything to do with healthcare, is actually the best and the highest level of service anybody can get. Service to patient is the highest level. In spirituality also, the highest level of service is service to patient. And we are fortunate enough to get this opportunity to do something for patients. So हमको तो ये opportunity by virtue of our studies and what we are going to do, by virtue of our profession, ये opportunity मिली है. Don't let it go waste and do best whatever is in your hand. World is waiting for you. Of course, this country is waiting for you, but the way we are developing and the leaders of this country is developing the India, the world is actually waiting for us to lead them. And you will see in next 10, 15 years that practically happening. So thanks a lot. And thank you for your patient listening. And I will wait for my first opportunity to come and take a class as an honorary professor in this campus. Thank you.
Thank you so much for your words of wisdom, sir. On behalf of entire organizing team, I would like to extend our gratitude to you, sir, for accepting our invitation and honoring us by being our chief guest today. It will be a great pleasure for us to listen you again in our future events too, sir. Now we proceed with our remaining felicitations. So I would call Dr. Havagire R. Chitme, sir, to come forward and felicitate our guest of honor, Dr. Arshad Khuru, Head Bioavailability and Bioequivalence Division, Sun Pharma, Gurgaon. A big round of applause, please. Thank you, sir. Now I would request Dr. Vinay Lathar, sir, to felicitate Dr. A.K. Singh, sir. We have reached to the end of inaugural session, so I would like to call upon Dr. Tanveer Navid, Deputy Dean, Health and Allied Sciences, Amity University, Noida, to come forward for vote of thanks.
A very good afternoon to one and all. It is indeed my proud privilege to propose the vote of thanks on this momentous occasion. First and foremost, my heartfelt thanks to our Honorable Founder President, Dr. Ashok K. Chauhan sir, especially his presence during this inaugural session simply spellbound all of us, and he is always a source of great inspiration to each Amitian. Thank you very much, sir, for your kind presence. My thanks to our dynamic Chancellor, Dr. Atul Chauhan, for always inspire and encourage us. Valuable guidance from Honorable Vice Chancellor Madam always inspire, encourage and motivate to do good initiatives, new initiatives, and this conference has no exception. And her presence during this inaugural session encourages us. So thank you very much, Vice Chancellor Madam. It is indeed a great pleasure and privilege for all of us to have a very gracious presence of Dr. Rajiv Singh Raghuvanshi, the DCGI, as the chief guest of this occasion. And sir, your speech was thought-provoking, but it was in such a simple word that it itself was a learning lesson from you. And it's a great pleasure for all of us. And moreover, you accept the honorary professorship at MIT Institute of Pharmacy. So it's a great honor for our institute also to have you as a honorary professor. Dr. Arshad Khuru, the very stalwart from the pharma industry. I know him as my senior at Jamia Hamdard, and over the last 3 decades, firstly associated with Renbexi and now Sun Pharma. Dr. Khuru contribute a lot of contribution, especially conducting the clinical trials, because he's the head of the bioequivalence and bioavailability division. Thank you, Asha sir, for your kind presence. I also like to thank Dr. W. Selvamurthy sir for his encouragement and presence during this inaugural session. In absentia, I would like to thank Dr. B.C. Das for all the support and the encouragement. My very special thanks to Dr. A.K. Singh sir. You are an amazing leader, sir, and during this conference, I realized it. And especially bringing the chief guest to this occasion is all your contribution. So round of applause for A.K. Singh sir for also. I also like to thank Dr. H. Chitme and Dr. Vinay Lathar, the new leadership of AIP, for all the support and encouragement to the organizing committee. For success of any conference, the role of organizing secretary is very important. And luckily for this conference, we have Dr. Navneet Sharma, who really comes out of ages and the proof is worthiness. So my compliment to you, Dr. Navneet. I also like to thank all my faculty colleagues as the members of this organizing committee. You really work hard and put up a great show. So compliment to all of you also. I also like to thank all the participants, the speakers, the scientific evaluator for this conference. I also like to thank the senior members present in this inaugural session, especially Dr. Captain Kapil Shukla, sir. Thank you very much, sir, for your presence and all other senior people. I also like to thank the STR department for their support throughout this conference, especially to Ms. Parul Singh and Ms. Harjinder Kaur for all the support. Thank you very much, and all the warriors, SNT warriors. I also like to thank the IT department, media department, admin department of MIT for all the supports. SERV, DST, and other sponsors, especially Redicon and AU Veda for their support for the conference. And as I personally realized, I have been associated with MIT last 16 years and part of many events. But one thing I realized that no event is successful without the support of the student volunteer. And for this conference, our student volunteer works day and night. I witnessed the SMS and the messages coming even the midnight hours. So hats off to you volunteers for putting this great show, and my thanks to once again to each one of you. Thank you very much.
So now I would request all the faculty members and student volunteer to come forward for a photograph. And after this photograph, we will be having our session from Dr. Arshad Khuru for the keynote session. So immediately after the photograph, we'll be having a keynote session by Dr. Arshad Khuru, sir.
Prachi?
Actually hungry for knowledge. So before I start my talk on bioanalysis, I have not kept it. Am I audible? I'm sure. So I'm also passionate about management development programs, and I have learned many things. In fact, delivered many workshops on effective communication skills. And in that effective communication skills, I'm also learning public speaking. So let me ask you one question. All of you, I'm sure you all are interested. That's why you're stayed back. Or maybe you're afraid of your teachers here. That's why you're here. So one question to students. What's the speed of tiger? Speed of tiger. Anyone? Anyone? Dr. Rajiv said that first person who will start question, then rest will follow him. Same thing is applicable here. So there has to be someone who will start the answer. Anyone? What is the speed of tiger? 50 kilometers. Okay. What's the speed of light? Anyone? Scientists. Okay, that's great. What is the speed of words? Speed of words which we are talking through our mouth. Anyone? Anyone? Sorry? Words per minute. It's 120. A normal human being can speak 120 words per minute. You know how much a professional speaker can speak? Any guess? Any guess? 180. 180 words per minute a professional speaker can speak. Now, last question. What's the speed of processing of words in our mind, in our brain? Scientists? Anyone can guess? How many words? Processing of words by our mind, by our brain. Last benchers, anyone from you? Usually I keep chocolates for these answers. Unfortunately, I couldn't get it, but next time I will get it. Anyone? What's the speed of processing of words of our brain? Usually a normal human being can process 500 words per minute. And a literate person can process 800 words per minute. Now, assuming that Dr. Raghuvanshi here, or I being here on the desk, or your teachers, when they teach you, or any public speaker, they can speak maximum of 180 words per minute. But our brain is able to process up to 800 words per minute. So there's a difference between what we speak and what you are going to process. What happens? Teachers, what happens? Nowadays, it's very common. Mobile phone or brain will start working on something else. It will start working on something else. The best way to, when you are doing public speaking or when you are teaching, or when you are discussing anything with anyone, please engage with your audience. Ask questions. Ask questions, have discussions, rather than having just one-way talk. That's the science of public speaking. Rather than having just one-way talk, engage yourself with speaker, and speaker should engage themselves with the audience. Am I clear? Sure. Thank you so much for that. Okay, let me begin this talk. I have not kept this very technical. I have kept it very non-technical. And how many of you know what's bioanalysis? Anyone who knows what's bioanalysis? I came to know that most of you are from pharma background. Bioanalysis. What is bioanalysis? Anyone? Okay, leave about anyone. How many of you doesn't know anything about bioanalysis? They don't know ABC of bioanalysis. You don't know anything about bioanalysis, so it will be very tough for me, but I will try to explain it in a very simple way. We are going to discuss what is bioanalysis, and before that, I'm also going to give you some idea about bioavailability, bioequivalence, what's absorption, what's distribution, what's metabolism, what's excretion, because that will help you in understanding how bioanalysis evolved in the world, especially in India, in the last 3 decades. And we are also going to discuss a little bit about future of bioanalysis. Bioanalysis is analysis of samples for quantification of drug or metabolite concentrations in biological matrices. When we are doing analysis of any sample, that's analysis. But when we are doing analysis in biological matrix, that's called bioanalysis. And biological matrix could be blood, it could be plasma, it could be urine, it could be your tissues, it could be tissue extracts. There's a difference between innovative development and generic development. I want to just give an introduction about how innovative development starts and how generic development starts. I hope every one of you knows what's innovative and what is generic. I'm sure. Generic, we usually call it as copycat. It's not actually a copycat. It's a generic version of innovative development. Both innovative and generic development, it starts with a need. The need is, it could be business need, it could be patient's need, it could be any need. The development starts with when our business development team tells us that, okay, there is a scope of this product or an indication in the market. Then there's an API development. In innovative development, there is then preclinical phase followed by clinical phases. In clinical phase, we have phase 1, phase 2, phase 3. After phase 3, usually there's an approval. And after approval, there's a commercial batch which is taken by the industry. It's then put in the market. And after the launch of the product, there is then phase 4. And in generic product development, we have again same thing. That's business development, business need should be there, followed by API development, then formulation development. Then we have studies called bioequivalence studies, which tells us how much drug is equivalent, whether the drug which is present in the body is equivalent to the innovative drug. That's called bioequivalence. After bioequivalence study, we get approval in the market, followed by launch. Now, why I'm telling you this? Because you will find these circles here in innovative development and in generic development. These circles tell us that in these things, in these particular development process, we need bioanalysis. Bioanalysis is very important in whole drug development process, as I have shown you in the slides. And we are going to discuss a little bit in detail that how is this bioanalysis evolved and what's the significance of bioanalysis. Before that, just quickly, for those who are not well aware of what's pharmacokinetics, pharmacology is divided into 2 branches: pharmacokinetics, pharmacodynamics. We know pharmacokinetics is what body does to the drug. Pharmacodynamics is what drug does to the body. Our bioanalysis is important in case of pharmacokinetics, which is absorption, distribution, metabolism, and excretion. Absorption happens when a tablet or a capsule, in fact, it happens through muscles also. But if we quickly see, when orally a drug or a capsule is taken, it goes into the stomach, dissolution happens, then absorption happens through either stomach or through intestine, small intestine. Now, this drug is present in the body because absorption has happened, and absorption has happened because of the different mechanisms which former students must be knowing about. Like there's a facilitated diffusion, there's a passive diffusion, there's an active transport. I'm not going in detail about that because that will be too technical. This is followed by distribution. When you might have seen if you just jump in the stream of river, what happens? You will get lost. Same way is when drug gets absorbed in the blood, it gets lost. So in order to not to get it lost, nature has made one mechanism. There are different proteins like albumin, globulin. These drugs get bound to these proteins so that they are distributed from the site of action, from the site of absorption to the site of its action. Then after metabolism, what happens? After distribution, what happens? Metabolism. Metabolism mainly is happening in the liver, and then finally excretion. Excretion mainly by kidney, but there are excretion happening. So these why I'm saying this, because this drug and metabolites, these are present in our body. It could be present in blood, it could be present in serum, it could be present in urine, and we have to analyze these drug and metabolite concentrations. That's why we call that as bioanalysis. And I want to just give an introduction also about what is viability. Viability is the rate and extent of absorption of a drug from the site of its administration into the blood circulation. And when we compare viability of two drugs, that's innovative and generic, that's called bioequivalence. And bioequivalence is very important in terms of regulatory requirements. In fact, in the morning also, I was discussing with Dr. Rajiv, and he was saying that I'm concerned about some of the centers doing bioequivalence studies which are not up to mark. These bioequivalence studies can be done using at least 4 types of assessments. One is pharmacokinetic endpoint study, then there is pharmacodynamic endpoint study, third one is clinical response, and fourth one is in vitro. Again, I'm not going in detail in that because that will be too technical, but I want to just give you one. That's pharmacokinetic endpoint study because this is related to bioanalysis. Pharmacokinetic endpoint study is when we evaluate drug concentration in the biological matrices using different techniques. Those techniques could be HPLC, could be LCM, SMS. We analyze if we are able to analyze those samples. Before that, if that particular drug or metabolite is able to get in the blood, then we call that as a pharmacokinetic endpoint study. Simple example is the amoxicillin, atorvastatin, simvastatin. Whenever we take it, these get absorbed into the blood, and that's why this is called as pharmacokinetic endpoint study. Even if we are able to estimate drug concentration in urine, that's also called as pharmacokinetic endpoint study. I will not go in detail, but I will just give you an idea about what's pharmacodynamic endpoint study. Like, for example, there's a molecule called orlistat. Orlistat is used, it avoids it to absorb, it prevents fats to get absorbed. So it's a local acting drug, but it does not get absorbed. How to do bioequivalence in that? We have to see some pharmacodynamic parameter. And accordingly, there's a clinical response where we do clinical trial. And fourth important, which I mentioned in my previous slide, is that's called in vitro studies. Like in case of sevelamer, we can do in vitro phosphate binding studies. And bioequivalence study has different components like clinical phase, we have the bioanalytical phase, we have pharmacokinetic phase, but we are, as I mentioned, we are right now discussing about the bioanalysis. Why is bioanalysis important? Anyone from here, anyone who has done any project or who has done some evaluation or had studied bioanalysis? Anyone from students? Your faculty was telling me that most of you are M-Pharm students. So anyone M-Pharm, have you done any bioanalytical project or have you studied about bioanalysis? Anyone from you? Girls are always better than boys in studies. Girls here? It's a fact. Girls? Not yet. Imagine that when we are analyzing those samples which we are collecting, like it could be blood. See, I'm not talking about the pathological labs wherein we estimate those thyroxine or hemoglobin. No, that's not bioanalysis. That's the clinical lab. That's the clinical lab separate. I'm talking of bioanalysis when a drug is taken and then we take blood or plasma serum and we have to analyze it with precision and accuracy. Now, if you assume that the method which we have developed is not accurate, what's going to happen? We are not going to have that proper development in place. Then assume that method is not reproducible. Now today we are analyzing some samples and tomorrow we are analyzing some more samples. What's going to happen? What's going to happen? Variation will be there. And then imagine if the method is not sensitive. Have you seen the pharmacokinetic profile? If the method is not sensitive enough, then we will not be able to characterize those parameters, which is usually AC0 to T and AC0 to infinity. We may be able to get some Cmax, but we will not be able to get AC0 to T and AC infinity, or even if we get it, it will not be characterized properly. Now assume that the method is not validated as per the regulatory requirements. Then again, there's a problem. That's why final analysis is important. If we quickly see some glimpse of regulatory guidelines, it clearly says that bioanalytical is important in terms of development process, and if the method is not rugged, reproducible, accurate, it will lead to failure in the product development or drug development. These specific statements are taken specifically from different guidelines. As Dr. Rajiv was mentioning about the business, and this is very important for you also. He was talking about whole pharmaceutical market of what India is going to do for export. But here you will see only bioanalysis. In 2022, there was a business of $2.9 billion US, and in 2027 it is going to grow to $6 billion US. $6 billion US. So anyone of you who wants to make a career in bioanalysis, you will be having a good career path. Bioanalysis has at least 4 components. First one is chromatography. What chromatography we are going to use. The second one is extraction techniques. Which extraction techniques we are going to use. Third one is detection. But above this, fourth one is guidelines. So we're going to quickly discuss how these have evolved in the last 3 decades, especially in India. And in bioanalytical, there are again 3 components. One is method development, where we see that how to optimize chromatographic conditions, how to optimize sample extraction techniques, how to optimize detection. Then it's followed by validation, followed by analysis of samples. And these are validation parameters. I don't have to go in detail. All of us know how medicine has evolved. Long back, many 1,000 years back, what was happening in medicine? Doctors used to give just some herbs. Then there was time passed. We started with extracts. Then time passed. There was some pills, big pills. Then time passed. Now currently we have tablets, capsules, injectables, and same thing has happened. Same evolution has happened in bioanalysis as well. If we quickly put a glimpse on chromatographic techniques, we have seen, you all are very junior enough, but in our times we have seen paper chromatography, we have seen thin layer chromatography, we have seen gas chromatography, we have also seen HPLC, and then finally we have also seen UPLC. That's ultra performance paper chromatography, which is evolved over a period of time. I remember time when I joined 1995 Ranbaxy. Ranbaxy at that time was considered as, anyone has heard about Ranbaxy? Students, not faculty. Ranbaxy. Where is Ranbaxy now? For how much money? I was actually part of Ranbaxy. Then we were bought. We were acquired. Sun Pharma was acquired by Ranbaxy, and now Sun Pharma is number 1 company. But during my times when I had joined Ranbaxy was number 1. In 1995, I was the first person in R&D who joined as graduate. Even though I completed my post-graduation and PhD after that, I was the first person who was recruited as a graduate in Ranbaxy R&D. And my colleagues or my batchmates, they didn't in fact believe it for next 2 or 3 days that I had been recruited by Ranbaxy R&D. Anyway, when I joined in 1995, what I found in Ranbaxy was I found out there were HPLCs. I remember I joined on Friday, and Sunday I went back. You must be knowing Shabir Rangwala was there. There was one of my senior. And as I told him that I joined Ranbaxy, first thing he asked me, Arshad, learn HPLC. I said, why? He said, this is upcoming. In India, you will find your career in HPLC. I said, there are multiple HPLC in Ranbaxy, so I'm in HPLC lab only. No, you should still learn HPLC. In 1995, I found out HPLCs throughout Ranbaxy, especially in my lab, there were 4 or 5 HPLCs. These HPLCs were mainly with UV detector and with fluorescence detector. That was in HPLC part. Then I also found out softwares were there in 1995, but those softwares were very basic softwares. Some of them were not having audit trails. Some of them we were not able to sync between two domains. Sometimes we had to even use manual auto samplers for injecting these samples. In extraction, we found out at that time there were at least 2 methods used. One was protein crash method. Protein crash and protein precipitation. You just put some agent. Like at that time, we were using perchloric acid, strong perchloric acid, or nowadays we are using acetonitrile and methanol, but perchloric acid or trichloroacetic acid was used for precipitation. And then we were also using liquid extraction. Then in terms of guidelines, in 1995, there were no guidelines. No guidelines for bioanalysis. There were only 2 or 3 papers. I remember one paper was from Midha and Shah. There was some other paper from Carles or someone else, which shows that how bioanalysis has to be done. No one in the lab, when I joined, was knowing what's bioanalysis. We were sitting and going through the papers and sitting on the HPLC, seeing that how to run the HPLC for bioanalysis. HPLC was very common, but in analytical industry, not in bioanalytical industry. What happened in 1996, 97? We started learning. I cannot say we became experts, but we started learning HPLC. We started learning bioanalysis. I remember time in 1996, we started working on projects which we called as pivotal or submission studies. We worked on some projects which were submitted, but most of the projects were outsourced to North America. In North America, especially in Canada, there was a very big CRO called Phoenix. And Phoenix was world-class CRO at that time, doing all the studies which were coming not only from India, but from across Asia and Africa. And there are also some CROs in the USA whom we were outsourcing the studies. But we, at least in those 2 years, we got an expertise in doing bioanalysis using HPLC UV or HPLC fluorescence. Remember, ours was the first bioanalytical center in India which was set up by Ranbaxy. There was some work going on at academic level, but not in the industry. Ranbaxy was the first one who started bioanalytical center in India. Even though we were now not so-called experts, we were not actually experts, but so-called experts, we were knowing what's bioanalysis, but there were multiple challenges. Multiple challenges in the sense that HPLC with UV and fluorescence, it is still having runtimes ranging between 15 to 20 minutes. And then at that time, cephalosporins were very common, and we were working on cephalosporins, where in CMAX usually is in the microgram. And it was very easy for us to do that. But the portfolio was increasing. There were some other molecules coming to us for development, wherein the sensitivity level was going to be down. There was a lot of pressure from the market to us that why can't you have turnaround time faster than what you are saying? Even though we were so-called experts in India, but we were lacking many things. We were not having LC-MS in our lab, mass spectrometry. There was only one MS, APA300, that's called APA300, in another lab, that's called analytical lab. But before that, also in between, we were not even having at that time to analyze samples using clarithromycin and pseudoephedrine. I remember 98, 99, when these 2 molecules came, because of low UV in these molecules, we couldn't use UV or fluorescence. But then we were able to use ECT detector for clarithromycin, and for pseudoephedrine, we were able to use GC. Anyone of you has used GC? What's the problem in GC? Main problem in GC? There's a major problem in GC. your sample has to be in a volatile form. Now converting liquid sample, because our sample is in plasma or serum. Now converting that sample into volatile form, it needs a lot of efforts. And then there are a lot of variations. We were able to complete, I remember pseudoephedrine was going on in our GC, and nobody in the lab was willing to work on that because they have to start the work at 9 o'clock in the morning, and at night, 10 o'clock, they used to leave the office. And what happened? There was one instrument called APH300 in another lab. And we came to know from our papers, from research papers, and from CROs to whom we were outsourcing that there's an instrument called MS, which can be used for bioanalysis. And we approached our management telling them that can we use this APH300. APH300 was in another department, and they were able to give it to us. We learned, at the time we learned MS, we were able to do some studies like simvastatin, enlapril, these we were able to do. But again, there were challenges. The challenges were there was still sensitivity problem. There was an ion source. If you might have read about LCMS, MS, or MS, wherein we have to do ionization. Ionization was a normal ion source wherein from morning to evening, we have to cut the capillary so that the flow of the liquid is such, and we can get ionization. Some of the times from morning till evening, every time a 1 meter capillary was cut by us. After every 30 seconds, we used to cut the capillary and see flow is there or not. Those were the challenge. But still, we were able to complete some of the important projects, and we were able to submit them again in non-US market. Concern for us and challenge for us was still US studies. And concern again for us was US was a lucrative market at that time, even though it is still there. But there were many projects coming up from US. Our management was telling us that why can't you do studies for US at in-house facility? But there were problems of audit trail. Audit trail was not very CFR Part 11 compliant. Then software issues were there. It was not connecting. There was no sync between LC and MS. We were having auto sampler wherein we were pushing the button, and then we are also pushing the button on the MS so that there's a sync. If the sync was more than 3 or 4 seconds, then we were getting errors. So those were all challenges we were facing. Then finally, in 1999, I remember there was a person called Umesh. Umesh was in AB Sachs. He came to our lab and he told me, Arshadji, I remember this time, Arshadji, I have good news for you. You don't have to cut the capillary now of ion source. I have got turbo ion spray for you. So I was shocked. I said that it's not possible. Since last 2 years, you are telling me to cut capillary every day for getting response. How can you say now you don't have to cut the capillary? He told me, okay, wait and watch. He put that ion source that's called turbo ion spray, ASI turbo ion spray. He put it in the MS. He told me to wait for 10 minutes to attain the temperature and inject. Response was 1,000 times what I was getting every day. 1,000 times. This changed our life. This changed actually my life. Because then after that time, for many months, I was going to my home early. Early means at least at 6 o'clock, 7 o'clock. Otherwise, it was always 10 o'clock in the night. This ion source, from ion source to turbo ion source, it changed our life, and it changed life of bioanalytical industry. With this also, in 1999, again, Shimadzu came up with HPLCs, which were very easily connected with MS. So turbine spray with MS, followed by different models from AB Sachs. We were able to complete US FDA studies for the first time in India. First time in India, we did submission studies for US, and in 2001, we were the first ones to get US FDA inspectors. This inspection was successful. What happened after that? You must be surprised that actually this was not an evolution, this was a revolution in India. Every pharmaceutical company, they started investing in bioanalytical centers. There came multiple CROs. Do you know how many CROs right now are in India? Anyone? Anyone? Small, big CROs? Nearly 1,000 CROs in India. But in 2000, there was not a single CRO. or in 1999, there was not a single CRO. Some CROs had come up like Pimta, Lambda, but after 2001, once we received first FDA inspectors and we successfully concluded the inspection, there was a revolution in biomedical industry in India. People started investing in CROs, pharmaceutical companies, they started investing in bioanalysis. And we also became training centers. We were getting people, they were learning, they were leaving us and starting their own CROs, their own centers. But then there were still challenges. Challenges were, again, here, one of the most important challenges was audit trail. Audit trail in those softwares at that time, even though there was a sync of softwares between LC and MS, but audit trail was still not CFR Part 11 compliant. And there was also sensitivity problems were still there because Sykes had launched some of the instruments, but we were not having those instruments. And here comes again the paradigm shift. There was a software change for LC. There was a software change for MS. There was UPLC, which came from HPLC to UPLC. Different guidelines came in the industry. This was again a total paradigm shift from one side, one state of non-regulated to fully regulated environmental bioanalysis. UPLC came in 2004. Anyone knows what's UPLC? What's the full form of UPLC? Anyone from here? UPLC full form. Ultra performed liquid chromatography. Do you know, I will tell you a story about UPLC. First UPLC in India was installed in Ranbaxy. आप लोग समझते हो ना Ranbaxy, a very important, a good company, but sometimes we are at a problem side also. Waters, they put free of cost one LCMS MS in our lab, but they were not knowing how to operate LC, UPLC. We were not knowing. They were not knowing even how to service UPLC. For I think 2 or 3 years, we were learning UPLC and they were learning UPLC from us in our lab. Interesting part is that after that, till today, we never bought UPLC from Waters. We never, believe me, we got UPLC, or that's called UHPLC from Shimanto, but never from Waters, because they used us as Unipig, as experimental tool. But it was a wonderful experience for us. Even UPLC also changed the bioanalysis. It became like, if you see runtime in HPLC with MS, if the runtime is around 6-7 minutes, with UPLC it can be over 2 minutes. If the liquid volume was 100 microliters, it's becoming 10 microliters. If injection volume was 10 microliters, it's becoming 2 microliters. If the solvent which was used was, for example, 1 ml per minute, now it is 100 or 200 microliters per minute, but we can understand how this HPLC to UPLC made a large difference in bioanalysis. There were different models then launched by AB Sykes. APA 3, then followed by APA 300, then followed by APA 3000, APA 2000, APA 4000, APA 4500, APA 5000, APA 5500, APA 5000, 6000, APA 6500, and now it's APA 7500. Now imagine the sensitivity levels from APA 3 or APA 300 to APA 7500. It is more than 1,000 times. Now, if you tell me to analyze at picogram levels, even 1 picogram per ml, some analyte, I will be able to do it in my lab. But 10 years back, even if you tell me to analyze nanogram per ml, it was a challenge for me. In fact, Waters is another company which launched multiple products from 2000 till date. The Quadra Premier, followed by then Zevo TQS. Even their instrument, the highest one, is able to analyze even analytes and metabolites at picogram level. In terms of, we just discussed evolution in terms of LC and MS, but there was also evolution which happened in sample extraction. I told you when I joined in 1995, there were 2 methods mainly used. One was protein respiration, other was liquid extraction. Before me, there used to be one dilution. But then what happened? After 2000, there came a method called solid phase extraction. Solid phase extraction, there were initially just some cartridges available wherein you can put the sample, it will hold the sample, and then you get rid of impurities, and elute the sample with some strong solvent or strong ester base. This became again a revolution when Waters came up with HLB, that is hydrophilic lipophilic valence cartridges. Every molecule, whatever molecule you put it through HLB, it will get retained. This again became not an evolution, which became a revolution. Today also, Waters HLB cartridges, it's considered as one of the best possible cartridge in the world. Even though there are multiple players in the market and they are coming up with HLB cartridges, but Waters has revolutionized this technique from coming simple C8 into HLB, that's hydrophilic lipophilic valence cartridge. Guidelines also evolved. One of the main important, as I told you, 1995, there was only 2 papers were there. But then in 1999, FDA came up with one guideline. In 2001, there was another guideline. Today, if you ask me, all the regulatory agencies, all, including India, Canada, Japan, Australia, New Zealand, Europe, every regulatory agency has their own guidelines for bioequivalence and bioanalysis, including US FDA. At our times in 1995, there was not a single guideline. And these guidelines have also evolved. And I must say, with confidence, that bioanalysis in today's world is one of the most regulated drug development process in the world. Most regulated. Now, question is, what next? It's as per, in my opinion, it's fully regulated. In my opinion, it's fully evolved. I don't need more than that. If you remember mobile phones, I will not tell the students the row in front here. We have seen evolution in mobile phones. I remember in 20 years back when mobile phone came, it was a huge set with 64 rupees per minute charges. Then it became 32. Then it became in 1 rupee. And nowadays I can say it's Jio has started free also. And in mobile sets also there were multiple changes that happened. We could not have imagined that no need of digital camera. We could not have imagined no need of laptops. Everything is now in mobile. Can you imagine now that evolution is still happening in mobile phones? Can anyone tell me that what evolution is happening in mobile phones? Anyone? Mobile phone के बारे में तो ज़्यादा पता होगा ना आप लोगों को. What evolution is happening in mobile phone now? What else? I want it from students. Please tell me. You know more than us in terms of mobile phones. You know, Apple is working on mobile phones which will not work on SIM. You don't need SIM. It will be on satellite. Any other thing? Flexible. What else? So it means even though we don't need those models, but still it's coming. Same thing is happening in bioanalysis. I'll just give you quick screenshots. What's going to happen in bioanalysis? 10 years back, I was in one of the conferences, and then as an expert in bioanalysis, I was being picked up by Waters. They asked me, Arshan, what do you want in future? 10 years back. I told them that I want a small mass spectrometry, which is of matchbox size. I want to put that matchbox size on any vein of the subject, give them drugs. This mass spectrometry should automatically withdraw samples, should automatically analyze it, and should automatically give me pharmacokinetic profiles. And sometime back, they told me that we are working on this. So it means there's evolution still happening in bioanalysis. What else? What else? It's in front of you. PK modeling and simulation. Now, regulatory agencies, they're coming up with, okay, lead about bioanalysis. Why do you need LCMS? You go for modeling and simulations. You have enough data, generate some data and simulate it, make some PKPP models and get rid of giving doses to different subjects or human beings. Third, but last but not the least one, very interesting one, AI. Artificial intelligence is going to change bioanalysis. How much? I don't know. I cannot tell you right now. I cannot even imagine what's going to happen and what's going to change. And I don't want to elaborate this. Maybe some other time we can discuss artificial intelligence. I've kept it only this much because I know there are multiple things happening in bioanalysis. And after 10 years or even after 5 years, we will see lot much more evolution happening in bioanalysis, which we cannot even think of. Same way, the way we are thinking about mobile phones. Just to summarize, I told you that Pharmacology has two branches, that is kinetics and dynamics. Then we have absorption, distribution, metabolism, excretion. We have 4 types of assessment. That's pharmacokinetic endpoint study, pharmacodynamic endpoint study. We have clinical response studies, and we have also in vitro. In case of pharmacokinetic studies, we usually need bioanalysis, but bioanalysis is also important when we do preclinical, because we have to do analysis in animal samples as well. Then we also discussed how LC evolved. We also discussed how MS evolved. We also discussed how extraction evolved. We also discussed how guidelines evolved. And we also discussed what could be the future of bioanalysis. I'm sure it will never be an end. You might have heard innovation is the mother of necessity, or necessity is the mother of innovation. This is applicable in both ways. Let's see in future what's for us in bioanalysis. Thank you so much for this. I'm sorry it was a little bit late and it was a lunch time, but I hope you might have enjoyed at least my journey with bioanalysis. Thank you so much. Any questions? Any questions? Lunch? You will get still lunch. I'm sure you will get lunch. Any questions? Yeah, please. Why shouldn't you? You should choose. No, that was no. That was a different. I told you that because they were working on us as a guinea pig, and we were not happy with them. We took a decision, not a scientific decision. It was a non-scientific decision that we will not buy waters from you, because you have worked. You were not ready with UPLC, and still you are wasting our time. That was it. It's every HPLC is same. Shimadzu UHPLC is also good. UPLC for waters is also good. You have to see actually the service support. It all depends on the service support. I hope so, but I cannot market on behalf of Shimadzu, but we have also Shimadzu in our labs. And you should also see spare parts. Some UPLC spare parts are a little expensive, but ruggedness in UPLC is much better than UHPLC. Any other questions? Yes, please. See, first thing is it is still invasive because you have to take blood. Yes, one thing is sure that you have to take less blood. But second most important thing is that it's not very new technique. We have been working on this since last 5-6 years. Unfortunately, this was not accepted by regulatory agencies. They have not. So they have categorically said that they don't have much confidence in this technique, right? Blood spot analysis. But you never know future. It depends on molecule to molecule. It also depends on positives and negatives. Now, positive in ST method is that sample is very clean, but negative is you need expertise and it's expensive. Positives in liquid extraction is it is very cheap, but negative is you need a lot of solvents. Solvents are expensive. These are carcinogenic. You can also use protein preservation, like using perchloric acid, acetonitrile, methanol, but only proteins get extracted. So it depends on molecule to molecule which extraction technique you have to use. You can use immiscible solvents for liquid extraction. But one important disadvantage for liquid, you shake it, everything will be moved from plasma to your solvent, including drug. So there's no control. See, there is no guideline for recovery. It has to be consistent. Even if it's 80%, it has to be consistent across the levels. There's no guideline which says that you need more than 90% recovery, more than 100%. Only thing is it has to be consistent. It cannot be at lower, your recovery is 100%, and at higher, it's only 50%. No, there is no guideline for how much recovery has to be. It has to be consistent. That's the guideline. And consistency means at lower, middle, high, CV should be less than 15%. 15%. Thank you so much. Thank you.
Thank you so much, Dr. Kuru sir, for your valuable insights on the international conference theme of therapeutic to biologicals. All these valuable insights gives us new perspective that one should adopt in their practice. Encouragement, enthusiasm, and dedication are the major aspect of research, and everyone should follow that. Thank you, ladies and gentlemen, for your time and patience. We are extremely privileged to have you all here today. We also appreciate your valuable time and patience you have shown us today. I would request everyone to proceed for lunch and be here at 2:15. We will be having our track 1 session in E2 seminar hall, just opposite to this. So please be here at 2:15 for track 1. So everyone please proceed for lunch. Thank you so much.
Hello. Yes, ma'am. I would like to request everyone to kindly be seated. A very good afternoon to everyone present here. On behalf of Amity Institute of Pharmacy, I would like to welcome our dignitaries present on the dais, our esteemed professors, and the outstanding members and our dear friends. Now we are going to start with the scientific session 1, which is on traditional scientific knowledge and phytopharmaceuticals. For this session, the chairs are Dr. Hemant Pandey sir, Professor Dr. Harsha Khadwal, and Dr. Mariam Sarwar. Now I request Dr. Harsha ma'am to introduce our first speaker, which is Dr. Hemant Pandey sir, Scientific Head and of Phytomedicine, which is Division of DIBER, DRDO, Uttarakhand. Ma'am.
So good afternoon, everyone. And it's my proud privilege to introduce Dr. Hemant Kumar Pandey, sir, who is Scientist F. Sir is presently posted at DIBER, Pithoragarh, and is heading Phytomedicine Division. He is actively engaged with different conservation and promotional aspects of Himalayan medicinal plants since last 34 years, and he has developed 7 herbal products, which is anti-leukoderma herbal product, which is Leucoskin, anti-eczema herbal product, Exoskin, anti-toothache herbal solution, advanced version of Leucoskin, that is anti-leukoderma herbal product, herbal health supplement, anti-UV radiation herbal cream, and antioxidant and immunostimulant herbal capsules through continuous R&D efforts on Himalayan medicinal plants. The technologies of first 4 products have been transferred to 4 reputed herbal pharmaceuticals of our country. 2 herbal products for the cure of leukoderma, that is Leucoskin, and for cure of eczema, which is Exoskin, has been launched into the market by ML Pharmaceuticals. In last 5 years, the total sale of Leucoskin is more than 100 crores. This has generated 3 crore royalty to the DRDO. This herbal product is a boon for leukoderma affected people, which is almost incurable skin disease and considered as a big social stigma in our country. The herbal product has given very good name and fame not only to DIBER but DRDO as a whole. He has filed 9 patents on these herbal products. He is author of 3 books, more than 150 research publications in different international and national journals of repute are in his credit, and presented research papers in more than 90 national and international seminar and conferences. He is life member of different scientific societies and recipient of a number of prestigious awards, including DRDO's most prestigious award, DRDO Agni Award 2005, DRDO Scientist of the Year Award 2020, DRDO Technology Day Award Year 2011, BioVet Fellowship Award 2014, Agri Innovation Award 2015, Laboratory Scientist of the Year Award 2016, Dr. Gorakh Prasad Vigyan Puraskar National Technology Award Year 2019, Fellow of Indian Society of Horticulture Research 2023, and Samarpan Samman Puraskar year 2023, and many more to come in coming years. We are feeling very proud, and especially sir myself, to get this opportunity to introduce you. So we welcome Dr. Hemant Kumar Pandey sir. Please sir. Acha. Okay. So, sir will be, the title of the talk is Leucoskin, a very effective herbal treatment for incurable skin disease leukoderma, developed by Diber DRDO Pithoragar. Thank you, sir. Thank you, sir. Please come.
Very good afternoon to all of you. The name of my institute is Defense Institute of Bioenergy Research. It is one of the sister institute of DRDO, doing R&D work on bioenergy and R&D work on Himalayan medicinal plants. The phytomedicine division of the institute is working on 2 fields: conservation of Himalayan medicinal plants and value addition. Under conservation, we have made extensive survey of Himalayan region, and medicinal plants, almost 250 medicinal plants of therapeutically important and economically important plants we have collected from Uttarakhand Himalayas. And after the proper identification, we have developed herbal garden in different climatic zones. Like plants of foothills, we are maintaining at our foothill station Haldwani, which is the headquarter of DRDO, Diver DRDO. And plants of river valley location and lower Himalayas, we are maintaining at our field station Pithoragar, where I am posted. We have 140 species of Himalayan herbs there in the form of live herbal garden. Altitude plants we are maintaining at our altitude station. It is at Oli, 9,000 feet. And almost 70 altitude plant species we are maintaining there in the form of garden. Aim to develop this herbal garden is mainly to conservation of Himalayan herbs, and to study the different plant growth stages of plant which are under threat of extinction, and again to create the awareness among the people regarding this valuable herbal heritage. Under value addition of Himalayan herbs, we have developed 7 herbal products. We have developed a herbal product for almost incurable disease leukoderma. It was our almost 16 to 17 years effort. And in the meantime, we have developed advanced version. After we further worked, we transferred technology of the Leucoskin anti-leukoderma herbal product to Emil Pharmaceutical. And Emil has launched this product into the market by trade name of Leucoskin. We further worked on this product for 5 years and developed advanced version, Leucoskin Mark II. And we have developed a herbal product for skin disease eczema, which is again difficult to cure. And after filing patent on that product, we have transferred technology to 3 herbal pharmaceuticals. And Emil has again launched our product into the market by the trade name of Skin in last year, 2023. We have developed herbal product for UV radiation cream. We have developed a anti-UV radiation cream. We have developed herbal health supplement, antioxidant and immunostimulant herbal capsule called Immuno Boost. We have filed 9 patents on these products. And after filing patents, technology of 4 products, anti-leukoderma herbal product Mark I and Mark II, anti-eczema herbal ointment, and anti-toothache herbals and 2-3 herbal pharmaceuticals. Leukoderma is almost incurable, as I already told. Some treatments are there, but efficacy is very less, almost 10 to 15%. In today's lecture, I am covering that part only. The title of my presentation is Leukoskin, a very effective herbal treatment for leukoderma, developed by Defense Institute of Bioenergy Research, Pithragal. Leukoderma, as you know, it is skin depigmentation disorder. Skin becomes white due to lack of melanin. Melanin is a protein responsible for coloration of skin. And it is considered as big social stigma in our country. The affected individual always remain in constant depression with the feeling of being socially outcast. And his confidence and self-esteem becomes very low. Since there is no satisfactory cure of the disease, so we took it as challenge and developed a herbal product for leukoderma. If you look at the definition of leukoderma, it is the disorder of skin in which melanin biosynthesis is disturbed either by destruction of melanocyte cells. Melanocyte cells are present on the basal layer of epidermis, which secrete protein called melanin, responsible for the coloration of the skin. Melanocyte cells or the tyrosine enzyme inactivity resulting white lesions on the skin. The worldwide incidence of leukoderma is 1 to 2%. In our country, the occurrence of leukoderma is almost 3 to 4%. Almost 5 crore people are suffering from leukoderma. In some parts of Gujarat and Rajasthan, its occurrence is as high as 7 to 8%. As per the extent of area of lesions, it is divided into 3 parts: leukoderma localista, vitiligo localista, vitiligo generalista, or vitiligo universalis. In localista, localized lesions are present on the body. It may be present on either hand, feet, or face. It is localized lesions. And in vitiligo generalista, almost 30 to 40% body is white. And vitiligo universalis, almost more than 90% of the cutaneous body is white, depigmented. It is if you look at the etiology of the disease, it is more common among the darker races, particularly in age group between 10 to 30 years. 28 to 30% cases, it runs in the family. It is genetic also. It runs in the family. If a person is suffering from autoimmune disorder like hyperthyroidism or adenocortical disorder, pernicious anemia, diabetes mellitus, alopecia, or psoriasis, he may be prone for leukoderma disease also. If a person is in regular contact with monomengyl ether of hydroquinones and phenols, chances of leukoderma are there. Deficiency of copper, vitamin B12, and aromatic amino acids also leads to leukoderma. Some problem with the इसके बाद ये इसको बैठा देंगे पता है वो यहाँ से हटा दो ऊपर से टाइटल आ रहा है एक्चुअली ऐसे ही डिलीवरी पे टाइटल ऊपर है टाइटल ऊपर इसको हटा दो इसको वहां से हटा नीचे रख दो नीचे theory of origin of the disease. chronic hepatic disorder. In 50 to 60% cases, what we have observed, a person is almost, the gastrointestinal problem is there in the individual, particularly constipation or acidity. Hormonal theory is also there. The middle lobe of the pituitary secretes one hormone, melanocyte stimulating hormone. Due to some shock or some other regions, if that lobe gets affected, it causes lack of that melanocyte secretion depletes and causes leukoderma. Neurogenic theory is also there. Some neurotoxins like phenols, quinones, hydroxyindols, and cuprous melatonin deposited at nerve endings. You might have seen leukoderma in tips of finger, where nerve endings are there, the fingertips, lips, tips of nose and ear. It is due to the neurogenic theory. It is autoimmune also. Autoimmune is response directed against the enzyme tyrosinase, because tyrosinase is the main enzyme in the melanogenesis, and if there is some problem in the in tyrosinase enzyme, it causes the leukoderma. And anti-melanin antibodies were also found in the plasma of leukoderma patients. Viral and mitotic infection is also responsible for the leukoderma. There are existing treatment of this disease: medical therapy, topical steroid therapy. Doctor advised corticosteroids for the treatment of leukoderma lesions, particularly dexamethasone, hydrocortisone, betamethasone. Ointment of these corticosteroids on the white spots. And topical sorolin UVA therapy is also there. Ointment of sorolin, isosorolin, 5-methoxysorolin, 8-methoxysorolin are available in the market. It has to apply on the white spots, and after 1 hour, exposure of UVA radiation, 320 nanometer to 400 nanometer. Oral UVA therapy is also there. Tablets, capsules, or injections of the sorolin are available in the market. And after consuming that tablet capsule, that after 2 hours, one has to expose that white patch in UVA radiation. Surgical therapies are also there. Skin graft, skin of pigmented area of the patient taken out and grafted on the depigmented area. Blister grafting, blisters are created on normal skin, pigmented area region, and depigmented area also. And the top layer of the pigmented area blisters were grafted on the depigmented area. Micropigmentation, tattooing. If white patch is present on the exposed body parts, it can be hide or concealed by the making tattoos. Autologous melanocyte transplant. In this, melanocytes are taken from the pigmented area of the patient, cultured in the lab, and by punch grafting, it is transplanted to the depigmented area. Adjunction therapy, depigmentation. If body is more than 80 or 90% white, so doctor advised for complete depigmentation of skin. Monobenzyl ether of hydroquinones, monobenzone is there in the form of cream and injection. It is applied on the hyperpigmented area, so it fits the color of that area, and that whole body becomes uniform in color, white. And some natural colors are also available to hide the white patches. But these therapies have number of side effects. The medical therapy causes shrinking of the skin and hyperpigmentation on the white spots. Topical UVA therapy causes severe sunburn, blister formation, hyperpigmentation, and hyperkeratosis on the white lesions. Oral UVA therapy causes sunburn, nausea, vomiting, abnormal hair growth, cataract formation, erythema or edema. Depigmentation causes inflammation, edema, itching and drying of skin. It is non-reversible also. Surgical therapy, all are experimental importance, restricted to the localized lesions. There is no answer if the disease is progressing. Overall efficacy is very poor. Almost 10 to 15% cases it is effective. This is the melanin biosynthetic pathway. It starts from aromatic amino acid phenylalanine, which hydrolyzes in liver and convert into tyrosine, p-hydroxyphenylalanine, which further convert to 3,4-dihydroxyphenylalanine, which is called DOPA, then dopaquinone, leucocompound, dopachrome, which on decarbonylation for 5,6-dihydroxyindole, and then it oxidizes into indole 5,6-quinone, which polymerizes to the melanin, a protein for skin coloration. Leucoskin has two components: ointment for topical application and oral syrup. Ointment is a polycomponent herbal formulation containing ingredient of 7 Himalayan herbs. Two plants are photosensitizer. Oral formulation contain ingredient of 5 Himalayan herbs. This is the product launched by Emil Pharmaceutical by the trade name of Leucoskin. This is combo pack containing ointment for topical application and oral syrup. The main biomolecule of the products are furanocoumarins. 4 types of biomolecules are present in our product: sorolin, angelicin, which is called 5-methoxysorolin, isosorolin, 5-methoxysorolin and 8-methoxysorolin. This is the HPLC chromatogram of ointment and oral syrup. This is the HPTLC fingerprint of Leucoskin, that ointment extract, oral dose extract, and the standard, which also validated the main biomolecule present in the product. The microelements in Leucoskin. Microelements play a very major role in the pigment formation, particularly copper, because copper is the prosthetic group of enzyme tyrosinase, which is the main enzyme for pigmentation. And zinc and iron has some role in the formation of pigmentation. This is the physiochemical parameter of Leucoskin, of ointment as well as oral dose: color, pH of extract, total soluble solids, specific purity, total minerals, conductivity, and acid insoluble as, etc. The animal pharmacological studies of the product were carried out from Indian Institute of Chemical Biology, Kolkata. They have made 4 groups of animals. The both side of the hair of both sides of the animals were clipped off. In one side, ointment was applied, and in other side, vehicle petroleum jelly was applied. In second group of animals, in one side, ointment was applied, and animals were fed with the oral dose also. In third group of animals, only ointment was not applied on the animals. Ointment was not applied, and animals were fed with oral dose only. Fourth group of animals were kept as control. Neither ointment was applied nor oral dose was given. Leucoskin ointment treated group of mice showing sign of pigmentation on the ointment treated area. In Leucoskin, in the area in which we have applied ointment, pigmentation started, and after 58 days, there was prominent pigmentation on the ointment treated area. The animals in which we have given oral dose also, and ointment was applied. The pigmentation started after 43 days, and 49 days there was pigmentation appeared on the side in which we have not applied ointment. It means oral dose is also working or also initiating the pigmentation. In control group of animals in which neither ointment nor oral dose was given, no pigmentation was there. Histological section of skin of mice in which ointment and oral dose was given. Longitudinal section. In treated animals, there was significant increase in number and size of hair follicles because the bulb of hair follicle is the site of melanogenesis. And in control group, number of hair follicles were very less, but there was significant increase in number and size of the hair follicles in treated group of animals. This is the cross-sectional view of treated and control group of control group animals skin. Similarly, same results were there. Significant increase in number and size of hair follicles in treated group of animals. MTT assay point was done with the help of MTTF assay on B16 F10 mouse melanoma cells, and at 24 days and 48 days, ointment was found non-cytotoxic, and same experiment was conducted for oral dose, and up to 200 mg per ml oral dose was found non-cytotoxic. We have done detailed toxicological evaluation of the liquid skin. Prime requirement of a herbal product is it should be free from heavy metals. In ointment and oral dose, no heavy metals was present. Lead, arsenic, cadmium, or mercury. The product was free from heavy metals. Toxicological studies we have conducted in-house and validated from Indian Institute of Toxicological Research, Lucknow. It is the recognized center for conduction of toxicological studies for acute, subacute dermal and oral toxicity in rats and mice, subchronic and chronic studies in rat and mice, mucous membrane irritation test in rabbits, skin sensation test in guinea pigs. At limit test dose, LD50 was found more than 2 gram per kg body weight. It is quite high. Dermal toxicity LS50 was found 1 gram per kg body weight. In 6-month exposure, no mortality and morbidity was observed. No morphological and pathological lesions on any vital organ or tissue. Organ weight ratio, no variation. And in hematological and serological parameters, no significant change was observed. It means product was safe for as far as toxicological parameters are concerned. These are the after taking due permission from the regulatory authorities, we have conducted clinical trials from Government Ayurvedic College and Hospital, Lucknow, a recognized center for conduction of clinical trials. These are the photographs. What we did before treatment photograph, and we took photograph in every 2 months, and regularly medicine was provided to the patients. This is before treatment photographs after 365 days. Before treatment photographs 340 days, likewise. That if a girl is suffering from leukoderma, her career comes under stake. So this is again hundreds of girls we have cured in our clinical trial. And this is before treatment 210 days, before treatment 270 days. These are the results of clinical trials. Male, female, it was divided, the clinical trial was divided into male, female below 40 years of age, above 40 years of age. In 51% patients, recovery was 75 to 100%. And in 21% patients showed recovery up to 50 to 75%. And 13 patients recovered 25 to 50%. Overall efficacy of the product was found 65 to 70%. We have screened the blood plasma of the leukoderma patients. Almost 40% we had taken for tyrosine enzyme and copper level, because the tyrosine enzyme and copper both are very important for this disease. what we observed in and compared with the blood plasma of the normal patient. What we observed in 15% patient, tyrosine enzyme level was not beyond detectable limit. In 60% patient showed lower level of tyrosine enzyme. 25% patients, though tyrosine enzyme was equal to the control group, but copper level was very low. In majority of patients, copper level was very low. In some cases, patients, copper level was equivalent to the control group of animals, but the tyrosine enzyme level was very low. So this might be reason of the white patches. Protocol for use of the medicine: gentle application of ointment once in a day from periphery to center, and oral syrup 50 ml with diluted with 20 to 25 ml of water for adult and 2.5 ml for children. Exposure in sunlight in morning and evening. Dietary restrictions: all sour food stuff should be restricted. Non-vegetarian diet, preserved and fast food should be restricted. Dietary suggestions: soaked gram is good for this restoration of this disease. Enough pulses. Vegetables like bottle gourd, bitter gourd, carrot are good. Advantage of Liquoskin: the product has no side effect. Being polycomponent, it is very efficacious. It restores autoimmunity and checks emergence of new spots. Cheap and easy mode of therapy. This is the launching photograph of Liquoskin in DRDO Bhavan. In last 5 years, total sale of Liquoskin was 100 crores. This has generated 3 crore royalty to DRDO. As I have already told, we further worked on this product and developed advanced version of Liquoskin anti-leukoderma herbal product Mark 2. We have made modification in ointment as well as oral dose, and some modification in process of manufacturing this product was also undertaken. And after modification, detailed toxicological evaluation was done. Clinical trial of the product was conducted from Government Ayurvedic College and Hospital, Lucknow. And clinical efficacy of the product was found of Mark 2 is 85 to 90%. It has enhanced. We have filed patent and patent has been granted to us. And there was the recovery time of the Liquoskin was 120 to 750 days. And in case of Liquoskin Mark 2, it has reduced to 90 days to 365 days. Time of recovery also reduced. We have, after filing patent, we transferred technology to Emil Pharmaceutical because Emil is manufacturing the Liquoskin. They have all techniques with them and all the facilities available with them. And this is the cover of the Liquoskin Mark 2, which is very soon this product will be in the market. Liquoskin Mark 2, advanced research of Liquoskin Mark 2. So what I can say, our product has changed life of thousands of people we have cured in our clinical trial. And company claims they have cured more than 1 lakh leukoderma patient. And still almost 1 to 1.5 lakh people are registered with the company. They are regularly taking medicine and getting benefited. And I feel myself fortunate. I have contributed in the development of this product, and this product has changed life of lakhs of people. Thank you very much.
Thank you so much, sir, for giving detailed, talk about Liquoskin, which is actually leukoderma is actually a social stigma people look down upon, and we should overcome that part also. And it's a like we are all thankful to you for developing this kind of product, where sir has given a detailed presentation about what are the symptoms, what should be avoided, and what all trials had been conducted, the existing therapies, treatment modalities, and also biosynthetic pathway of melanin synthesis. the physico-chemical complete details about leucoskin. So it's a great learning for all the students of, B Pharma, M Pharma. In future, you can also develop, you can learn from sir. So the floor is open for question and answer. Because of paucity of time, we'll take one question. So anyone can ask any question. It's a wonderful opportunity. Yeah, please ask.
Welcome. Suppose a person gets recovered, but he has to follow the dietary regulations for next 6 months. And what we suggest, he should not stop taking oral syrup. Oral syrup he should continue till at least for 6 months. Then there won't be chance of relapse.
So
Thank
much,
you.
sir. Thank you, sir.
Thank you, sir, for this very informative session. Now, I would like to request Madam Harsha Khadarwal to welcome and facilitate Hemant Pandey, sir. Can we have a big round of applause? Thank you, madam. Moving ahead, I would like to request Dr. Mariam Sarwat, madam, to introduce our next speaker, Dr. Suhail Parvez, sir. Thank you.
Good afternoon, everyone. It's my pleasure to introduce Dr. Suhail Parvez, sir. He's a professor of Department of Toxicology and Dean School of Interdisciplinary Sciences and Technology, Jamia Hamdard. He happened to be my senior also, as I have also completed my PhD from Jamia Hamdard. Dr. Suhail Parvez has done his MSc and PhD in toxicology in 2003 from Department of Toxicology, School of Chemical and Life Sciences, Jamia Hamdard. And he did various postdocs from Department of Neurology, Germany, and from Laboratory of Cell and Molecular Signaling, Queen's Medical Center, USA. And then he has got the prestigious Humboldt Fellowship and has worked as Department of Neurophysiology, also in Germany. And he has got another fellowship from Royal Society of Biology, UK. He has been actively engaged in research focused on basic life sciences with application in translational research during the span of last 2 decades. He has published more than 200 reputed research papers in peer-reviewed PubMed and Scopus Index journals with an average impact factor of more than 5. His H index is 48. Total citations are more than 6,000. And I-10 index is 71. He has completed about 15 research grants from international as well as national extramural funding agencies. He has guided more than 40 PhD research scholars in supervisor as co-supervisor and has been awarded their degrees. Dr. Parvez in University Coordinator Program for PERS and for FIST as well as Stuti Program from Department of Science and Technology, Government of India. Over the years, Professor Parvez has successfully imbibed an expertise in both in vitro and in vivo models and used to explore basics of neurosciences. He has extensively worked to explore the possible therapeutic interventions of nutraceuticals and their mechanism for the treatment and prevention of several neurodegenerative as well as neurological disorders. He is also working towards the drugs repurposing for standardization several neurodegenerative in vivo models in his lab. The rodent model of traumatic brain injury, behavioral tagging, and in vivo electrophysiology developed by his research group are the first and only in the entire country. His lab has also successfully established other disease rodent models such as ischemic stroke, Alzheimer's disease, and subarachnoid hemorrhage. In addition to rodent models, he has also been working extensively on development of alternative models to gain the mechanistic insights of the mechanism of neurodegenerative disorders such as Alzheimer's and Parkinson's. He has been working on Drosophila model to screen the potential drugs for neuroprotective effects in Alzheimer's. The C. elegans model has also been established recently in his lab with the purpose of screening of possible therapeutic targets for neuroprotection in Alzheimer's and Parkinson's disease. He has a state-of-the-art laboratory with all these model systems. I have personally visited it, and it is very fascinating. So I welcome Professor Suhail Parvez, sir, to deliver his lecture on the targeting mitochondria and Alzheimer's disease. Sir, please.
Light changes, huh? So, we have this, and we have this. Okay, go back. Let's go minimize. स्क्रीन शेयर करते हैं नहीं
It will be on share mode.
स्क्रीन शेयर
Screen share mode. Share screen. Teams को open करो.
seconds. Good afternoon, everybody. It's been a privilege for me because I am visiting Amity Institute of Pharmacy for the first time. I have been a regular visitor at the Institute for Neuroscience at Amity. Almost every month I come. But for this particular institute, it's the first. I would like to thank Dr. Navneet, who has invited me here, and also my institute mate, Professor Mariam, who has given such a detailed, elaborate introduction of me. So, as I represent Jamia Hamdard, so I have to first do my duty and work as its ambassador. So, how many of you here have heard about the name Jamia Hamdard? Okay, as I can see, only less than 50% of the hands up. You are going to have the next slide where I tell you about the history of Jamia Hamdard. So this institute actually was the dream of our founder, Hakim Abdul Hamid, who started it as an institute of history of medicine and medical research in the 60s. Interestingly, Unani medicine, pharmacy, nursing, and Islamic studies, these four disciplines were part of Jamia Hamdard with the erstwhile University of Delhi. We were affiliated to that. Due to the efforts of the then Prime Minister, Sri Rajiv Gandhi, in 1989, we got the deemed university status
And in 2011, we actually got our medical college and the Center of Excellence of Ayush in 2022. And obviously, when I am talking about pharmacy and Jamia Hamdard, I have to mention the NIRF ranking and how it goes over the years. And we hope for the best also for this year, not only for Jamia Hamdard, but also for Amity. Okay, so today I'm going to talk a little bit about Alzheimer's disease. The problem is the kind of audience I have are mainly students. So the attention span is exactly like a kindergarten. Why? Because of so many gadgets we have. Everybody has free access to phones and Chehre ki kitab and WhatsApp. So something has to be different in the lecture which will attract or maybe increase the attention span. Okay, let's see. So when I talk about neurodegenerative diseases, technically in our country, we identify it as Alzheimer's, भूलने की बीमारी, Parkinson's, हिलने की बीमारी. Apart from that, when you go to the third one, then we miss. Why? Because if I say Huntington disease, हाँ वो कुछ होता है. So, as this slide, which is the highlight of today's lecture, will be very interesting for you, and as you are all in deep slumber after this nice lunch, let us wake you up. So, I'm going to point at every frame, and I would like to listen in cohesion from you the names of the movies that I'm going to point out. Okay? So, wait. I have to first mark it up. Okay. Now everybody has seen the movie, but the voice is only coming from 10. So if you have spent so much of money, it is required now you come up with the names of the movies you have watched. Chalo, fir se. Next. 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 दिख नहीं रहा इसलिए ब्लैक है. Next. चली नहीं इसलिए आवाज कम है. Now. Let's take into a different dimension. You have spent 3 hours watching the movies. You have invested minutes, hours, days listening to the songs. I'm going to ask you only one question about these movies. All these movies are actually based on neurological conditions. So now what we will do, with the same intensity, with the same interest, we will scream out loud in unison the conditions of these movies. So let's start one by one. First, next, Shanti. सारे पैसे बर्बाद कर दिया. Next, आया बेट वो बात नहीं थी. Next, सिर्फ एक के पैसे वसूल हुए. Next, गुड. किसी ने अल्जाइमर्स नहीं बोला, मैं इसी पे बोलने वाला हूं. नेक्स्ट, गलत जवाब. चालान कटेगा इनका. So, just imagine, we, students of pharmacy and biological sciences, will tomorrow be in midst of audience, our own children, our siblings, and then they will ask you, what's the name of the movie? You will say, Amir Khan, Tare Zameen Pe. What's it about? हाँ वो कुछ बच्चों में होता है. This is the problem, this is the issue. And this really needs attention. Why? Because this is something that has to be integrated in things. For example, when we talk about Alzheimer's, Alzheimer's is not the whole thing. Technically, Alzheimer's is part of a big umbrella of dementia. Dementia is the bigger umbrella, and Alzheimer's is contributing the maximum to the umbrella of dementia. So, when we usually see, we see a healthy brain that does not have enough beta amyloid plaques. But the Alzheimer's brain, it will have much more amyloid plaques. So we say, okay, because of the amyloid plaques, we have too much of things that are going on, which is then causing the Alzheimer's disease. This is the global scenario. And if you look at the Indian subcontinent, which is, I have to get the cursor, which is here. So you see, we are one of the leading countries because the countries which are orange or dark orange are the ones which has more numbers. Now, every 2 out of 3 people will be having dementia in their future course of life. That is bad news for the youngest generation. Why? The life expectancy is rising like anything, and we are losing out to the demand of technology and modern day, and we are compromising with diet, with sleep, with health, with practically everything. So, we need to understand that in Alzheimer's, actually the problem lies in the entire diagnosis. If you look at Alzheimer's, it actually happens in different phases. And in every phase, a different part of the brain is affected. And only a clinician can tell us that we are suffering from which particular area of the brain. So this is an important part because these will be at different stages of the progression of the disease, and at the same time, it will lead to the different pathophysiology as well as etiology. Now, when we look at the global market, the entire Alzheimer's disease treatment is focused on 2 different scenarios. One is the NMDA antagonist agonist, and the other is the ACHE inhibitors. So most of the drugs, whether it is donepezil or it is rivastigmine or anything, they are actually focusing on these particular pathways of NMDA and acetylcholinesterase. So the idea is that is Alzheimer's only caused due to amyloid? No. There is a non-amyloid pathway too, and there is a more acceptable amyloid pathway too. So what we try to do in our lab is to follow the amyloid one. And if you are a student of pharmacy, if you have been following the recent drugs that are launched in the market, Edulham was launched in the year 2021 as the next drug for treating Alzheimer's. Okay, so there is this nice concept of drug repurposing these days. Why? Because it branches out the real time that is taken for a drug development. You shorten the time. So, I skip this part and I skip this too. Okay. So, what we did, we took dopamine D2 agonists. If you look at the definition of Parkinson's disease is usually loss of dopaminergic neurons in the substantia nigra, and we tend to refill the stores of dopamine with this dopamine agonist. So there are 2 agents, ropinirole and pramipexole. They were introduced almost 2 decades back, and till today, they are believed to improve motor scores in Parkinson patients. So what we did, patch clamp technique developed by Neher and Sackman, they actually developed this technique to measure ion channel. And they got also the Nobel Prize for it in 1991. So what we did, we took the mitochondria of the brain and we tried to actually measure the permeability transition pore. So we isolated the mitochondria of the brain, We then made it swollen and we broke the outer mitochondrial membrane, and the inner mitochondrial membrane was patched with a glass pipette, giving us the activity of the permeability transition pore, which looks something like this. The idea is, due to ROS, due to oxidative stress, due to increased phosphate concentration, the permeability transition pore is leaky. Then the cytochrome C, which is safe within the mitochondrial compartment, is getting out in the cytoplasm. Once it is there in the cytoplasm, it is activating the different caspases. And once it is there, then the apoptosis is on the way. We wanted to see whether this particular drug can block the PTP opening, and by patch clamping we could show that yes, they are actually blocking, and when you take away the application of the drug, the channel is opening up again, making it a pharmacological reversible action. Okay, so we wanted to try this in a rodent model, and what we did was we took the rats, we injected them with beta amyloid, which is through the ICV route. For that, you need a stereotaxic apparatus. And once we did that, we infused in each of the two lobes the beta amyloid. Protocol says that within 21 days, because of the infusion of this beta amyloid, you will have beta amyloid fibrils. There is a specific protocol also for forming the beta amyloid solution, which you need to expose it to the rat. So there is a surgical procedure by which you infuse it, and within 3 weeks, it is expected that the beta amyloid plaques will be formed in the rats. So when we looked at the results, we did it with two different concentrations, 0.5 and 1 milligram of pramipexole. The results that you are seeing now are of the Morris water maze. So you can see that in the control, there is a very small line here. That means the movement of the rat in finding a platform. Then in the beta amyloid group, there are too many lines going round and round. That means the animal has difficulty actually in finding the particular platform. Whereas the low concentration and the high concentration, you can see that it is improved. It's not exactly like control, but it has improved significantly. So that was the idea. So we did some supplementary experiments, for example, getting the ROS production of mitochondria by FACS, getting the mitochondrial membrane potential done, because membrane potential actually goes down, that makes the PTP open. So we did membrane potential experiments. We did OXPHOS, oxidative phosphorylation, where you measure all the different complexes by Western blot. You don't do it with an oxygraph. You do it with a Western blot. And very, very interesting experiment was to isolate the hippocampus and to isolate mitochondria of that hippocampus and then look at the cytochrome C expression. I don't know if it is visible to you. Yeah. So you can see here, I have on the left hand side the cytosolic fraction and on the right hand side the mitochondria fraction. So it would mean that in my beta amyloid group, you can see that there is a big blotch in the cytochrome panel, in the cytosolic panel. Okay, it's here. You see here. Compared to the mitochondria. the second lane of cytosol and the second lane of mitochondria. You can see that the cytosol has a bigger blotch. That means most of the cytochrome C has leaked out from the mitochondria into the cytosol. But when you look at the protection groups, it is different. So now this was the major experiment in convincing the reviewers that our drug pramipexole was good in preventing the leak of the cytochrome C from the mitochondria into the cytosol. And this actually helped us publish in the Journal of Neurotoxicology. Mohammad Salman actually is the first author in this, who is currently a postdoc in the University of Tennessee. Now, basically, our current treatment regime is focused on, as I said, the NMDA receptor antagonist, and the acetylcholinesterase inhibitors. So, whatever we do, however we go about it, we cannot change the mechanism. So, the idea becomes you synthesize something, you change the moiety, you do docking, you do MD stimulation, and then with everything you come up, okay, this is 5% better, this is 10% better. I feel we need to imbibe certain things to fill this translational gap. Why? Because there will be hundreds of compounds that are available. The chemists, the pharmacologists will definitely come up with more and more alternatives. But usually, if we again only target the NMDA as well as the acetylcholinesterase, you have no other way out. It's only this or that. So, just to share with you, our Indian traditional system for Unani is very rich. And what we have, a drug known as Majune Falasva, Majune Brahmi, and Majune Baladur. These three Unani formulations have been there for many, many years, and they have been used for the Unani concept of neurological disease. So technically, you have Nisyan. Nisyan means something which is affecting your internal system of the mind. And this whole Unani concept is about three different quarters: faculty of memory, faculty of thought, and faculty of imagination. And at that time, when neuroanatomy was not there and neurophysiology was not there, it really captured that how different portion of the brains actually regulate different functions. So we took one of these, which is Majune Falasva. We didn't isolate anything. We used the whole compound and we calculated the dose of rats from the human dose, and we tried to see some things. For example, first experiment was designed to do in N2A cells, where we did bright field imaging, where we did cell proliferation, and we also did the Ross induction. So N2A cells is usually described as a better alternate for studying beta amyloid induced Alzheimer's like symptom in comparison to SHY-SY cells. Whenever you will hit PubMed, you will always get SHY-SY cells. So based on the initial experiments of Majune Falasva under in vitro conditions, we then did the in vivo experiments with these. So these are again the in vitro ones where we have the NF-kappa B and the other fluorescence ones, which I skipped. And then we did the in vivo experiments where we again took two different concentrations of Falasva and we did elevated plasma study. This is again a long-term memory test. These blue dots that you are seeing, these are actually the heat maps that are generated by the Animus software, which takes control of that. And our main target was to show how exactly the neuroinflammation is being catered for, which is one of the major effects in Alzheimer's disease. So we looked at the Krebs BDNF, and we also looked at the ROS with the DCFDA fluorescence. We looked at the biological macromolecules of protein through the protein adduct formation of 4-HNE, the left-hand side panel, and also the different panel assays, and finally the hippocampus C1, CA1, CA4, and dendrogyrus region. So all of this gave us this particular idea that CAM kinase 2 and Krebs are significantly modulated by our philosopher. So rather than saying that modern medicine is the best or the traditional medicine is the best, we need to have this brilliant combination of two things at the dose that will not be toxic, which can cater for not one but bucket of the effects that are there. So to finish off, I have two more slides for you, which is pretty interesting. The first one was published last year in Nature Aging. Just imagine the power of artificial intelligence. They are using a digitized tool with a phone app to check whether you are having an early diagnosis of Alzheimer's. Imagine something like that happening. The second one, again, a very recent paper from Lancet, where they have made a tool by which they will scan your eyes, and they have developed tools. Scanning your eyes will give you markers for you to assess whether Alzheimer's symptoms are there or not. Now, this is something that should excite you as an entrepreneur. You don't need to be a doctor. You don't need to do PhD. You don't need to invest so much of your time. You need the right balance where you can imbibe something like this at an early stage and come up with something that can really change. So these are the funding agencies that have helped me over the years, and I have been very, very fortunate to have a very big lab of almost 22 people who are working all through the time. And Salman was one of the scholars that I showed you, who is not in this frame because this is a recent picture. And the Unani experiments with Falasfa were done by Pinky, who is in the CCRM project that we have. So these are the collaborations that have helped me over the years, and thanks a lot.
Thank you, Suhail sir, for your outstanding talk. And that's such a sophisticated experiments which you have done, we can only imagine in our dreams. No doubt you have published such nice papers in interesting journals like neurotoxicology. So the beginning of the lecture was tantalizing, and it surely has woken up many delegates from their slumber. So can we expect some interesting questions from the delegates? Any questions?
Sir, first of all, it's a really wonderful piece of work. and I also worked on Parkinson's disease. So I have a few doubts here.
Yes, sure.
So
Go
you
ahead.
told me that you have taken pramipexole and
Ropinirole. Yes.
But what made you choose only these drugs? Is there
Yeah.
any?
So what happened was, when I was working as a postdoc, and when these two drugs came into the market, everybody was thinking that the action of these is better than dopamine or L-dopa, that is giving the effect. So when groups started working on it, only very later they realized of all the dopamine agonists, these are specifically functioning on the autoreceptor 2. So these are dopamine D2 agonists. So the major reason for me to take it was their direct interaction with the permeability transition pore. Many people are targeting many things. For me, my lab is working on mitochondria. And as all the experiments that were designed in and around mitochondria, the major reason of using pramipexole and ropinirole was that they are neuroprotective. There is no doubt about it. They are wonderfully interacting with mitochondrial PTP. There is no doubt about it. And nobody tested dopamine D2 agonist in Alzheimer's. So the idea was whether this is good enough not only to work via the channel of mitochondria, but also give corroborating evidences that will say, okay, this is there. But yes, we are at a very nascent stage. It's only 1 or 2 studies that have been there. So a long way to go.
Yeah, one follow-up question, sir. So
Sure.
how can you rely the result which you obtain from the SHY, the neuronal cell line? Because it is very, very general kind of cell line.
I didn't use SHY-SY. I have shown N2SLs. That was the reason I was telling you.
Okay.
I was telling you why we should not rely more on SHY-SY because I didn't use it. I use N2SLs.
Thank you.
Yes, please.
In the first slide only, you have shown that India has more prevalence in this Alzheimer's. So what can be the reason behind that?
There are many reasons behind them. I will just show you one slide. Do I have time for 2 minutes just to show her?
Yeah, I am Aradhya Jaiswal from Kirori Mal College, University of Delhi.
Can you see this slide? One minute. Is it better now?
Yes.
Now, our problem is if we have an arthritic pain, we will try to walk and make it go away, accepting the limpness. If we have diabetes, we will tend to go to sugar free or we will have chai and insulin shot at the same time. But if you have Alzheimer's, nobody else is in problem but the patient himself. Just look at this data that has been generated only last year. 28% people believe if the doctor tells you that you have Alzheimer's, they say, I might get an incorrect diagnosis, so I will not go to the doctor. I have been suffering from presbyopia-like symptoms for the past 1 year. मुझे दिख नहीं रहा था पास का. But what I did, and now it's 1.5. So Alzheimer's, for us, it's a problem Because until and unless you have any problem related to the heart, everything is fine. Why? Because we are accustomed to stent lag jayega, bypass ho jayega. Alzheimer's, ये तो आने जाने वाली बीमारी है. And the reason again is the amount of stress that we Indians are carrying from job to home to work everywhere, and the kind of lifestyle we have. We sleep with our phones, we wake up with our phones, including you all. If I tell the younger generation for the next 1 week, you are banned from Facebook and WhatsApp, many of you will fall into depression and maybe even hospitalized. So that's the real story behind it. And of course, we are vulnerable. We never go for checkups. Even if we go for checkups, we will go to a path lab. Cholesterol check करा दो, glucose check करा दो, H1B AC check करा दो. ये कभी नहीं कहेंगे ये check करा दो. Thank you.
Thank you, sir.
Thank you, sir, for this very informative session. Now, I would like to request Madam Mariam Sarwat, Dr. Vinay Lathar, sir, and Dr. Tanveen Nave, sir, to felicitate Dr. Suhail Parvez. Can we have a big round of applause for them? Yes, sir. एक बार सर पीपीटी चालू करते हैं. वो कर दो मैं. क्या सर? एक सेकंड सर. Thank you, sir. Moving further, I would like to request Madam Swati Madan to introduce our next speaker, Dr. Sanjay Sharma. I would like to request Dr. Harsha Khadwal to introduce our next speaker, Dr. Sanjay Sharma.
It's okay. It's okay. So I'm Dr. Harsha Kharkwal, okay? That name is. So good afternoon again. And I think what you have learned from last lecture, I think for 40 minutes from now or 1 hour from now, if you can just switch off or put your phones on silent mode and test yourself by listening to the lectures more, that will be very, that will be testing your patience also. Okay. So, it's my proud privilege again to welcome Dr. Sanchit Sharma sir, who is founder and director of AMIL Ayurveda Limited and executive director also of AMIL Pharmaceuticals India Limited. And as you all know, AMIL, a renowned name of quality herbal health product in market, established with aim of taking the legacy of Ayurveda ahead scientifically since 1984, and it is the leading health contributor to the society by innovations, developments, manufacturing, marketing, and promoting a broad range of Ayurveda products in different dosage form indicated in several subacute and chronic disease conditions. So, Dr. Sanchit Sharma sir has done his B Pharma, M Pharma, and PhD in pharmacognosy and phytochemistry from Jamia Hamdard University. He is an experienced leader with a demonstrated history of working in the Ayurveda, herbal pharmaceutical industry, strong business development professional with a doctor of philosophy focused in natural product chemistry and pharmacognosy. He is experienced in leading sales and marketing team along with skills in herbal product development, management, market research and management. As a researcher, he has worked on various plants mentioned in Ayurveda to scientifically establish the safety and efficacy. He is currently working hard to research out new Ayurvedic formulations to provide effective treatment to diseases like infertility, chronic skin diseases, CKD, etc. In the past 2 years, Amil has developed hundreds of personal care products under brand name Amil Ayurveda with the mission to help 1 million people switch from harmful chemical based personal care products to safe and wonderful Ayurvedic products. Ayurveda has become a fast growing company and is ranked upon top 100 D2C brand of the year 2022. Dr. Sanchit has successfully participated in business meet of Indo-American Wellness Conclave, which was held in Santa Claus, US, and he established the business relations there. He has designed and developed commercialization of polyherbal formulation, Neeri KFT as kidney functional toner. He has established Mangostin, a value addition of polyherbal formulation, Produle to combat acne, and management and marketing strategies development to utmost customer satisfaction in rural as well as in urban society. He has won several awards like Marketing Excellence Award for BGR 34 by AIOCD Mumbai, then Recipient and Recognition Award at Kuntala 2018, a national seminar on Ayurveda and trichology at Hyderabad. Honored for professional participation in the largest class of Charak Samhita organized by NASAYA in collaboration with Vibha and Ministry of Ayush, Government of India in 2018. He was honored and and also had participated in national conference at NEMA 2018 at NMDC Hall, New Delhi. So we welcome you, sir. And the title of his talk is Novel Herbal Pharmaceuticals for Drug Delivery. Dr. Sanchit. Yeah. Thank you.
Thank you, Dr. Harsha, and thank you to the organizers of the conference, and thank you, MIT University, for inviting me for the talk. And good afternoon, everyone, and to all the students here and all the professors and seniors sitting in front of us. So my topic is novel herbal pharmaceuticals for drug delivery, and we have already had two very informative sessions by Dr. Pandey on leukoderma and Dr. Suhail also on Alzheimer's. And I think I just want to know the type of gathering we are having here. We are all undergraduates here or postgraduates, pharmacy graduates, MSc students also, sir. So as we know that the herbal pharmaceutical industry is growing very rapidly, and since Ministry of Ayush has been formed in 2016, 15 and 16. Since then, under the leadership of our Honorable Prime Minister, Ayush industry is growing rapidly. And in 2020, when Corona had struck, since then, people are getting more aware about the Ayush products. So, since we know that the herbal industry is growing at currently in 2020, what we had, the data we have, it is around 1,50,000 crore market size of the Ayush industry that we are having here. So phytopharmaceuticals is still in the native stages. Majorly, the market is from Ayush, which is containing Ayurveda, Yoga, Unani, Siddha, homeopathy, and alternative system of medicine. So all including this contributes into the Ayush market. So today the topic is the novel drug delivery system on the herbal drug delivery, and as we all know that plant have the nature remedies and have been used by human beings on earth since the ancient times, and our Ayurvedic or the ancient texts refer how effective the herbal drugs are, and now herbal medicine have gained worldwide popularity, and treatment to each disease is hidden in nature. As we know that Dr. Pandey also recommended that vitiligo, which was once incurable, he has found a treatment for vitiligo. Similarly, for CKD, which was not curable at one stage, herbal medicines have cure for a lot of diseases that are there, and that has given us a scope for each one of us, whether we are in going to do the research, whether we are going to market the herbal products, or whether we are into the any aspect of the herbal drugs. The side effects are lower, as you know that the side effects of the herbal drugs are low. The effectivity of the products are very good, and the lower cost. So herbal drugs are the drug of they are gaining popularity, and we know why, because of the following reasons. And the need for a novel drug delivery system is that the basically in the recent data reported is that more than 70% of the new drugs formulated are showing poor water solubility, which becomes a limiting factor in the absorption of the drug after the oral administration. The limitation in the bioavailability of the natural products, active components includes poor solubility of the ingredient, poor stability, and then the colonic acidity and poor metabolism by the effect of the gut microflora, poor absorption across the intestinal wall, poor active efflux mechanism, and first pass metabolic effects are among the factors that make the failure of the clinical trials. So as we know, the herbal drug industry is growing, but at the same time there are challenges with the absorption and with the bioavailability at the target site of the in the body. So that is why the need for new drug development, the novel drug development delivery system is required, and that is why we have seen there is a keen interest of the market for the novel drug delivery system. Now, in the novel drug delivery system in herbal pharmaceuticals, nanomedicine has recently earned enhanced attraction for its ability to efficaciously diagnose and treat various ailments. Various approaches in cases of novel herbal drug delivery system includes different types of formulations such as liposomes, ethosomes are there, phytosomes are there, microspheres, transferosomes are there, nanoparticles, microspheres, ethosomes, transdermal drug delivery systems, and different so on have been developed in the cases of NDDS in the herbal pharmaceuticals, and employment of innovative drug delivery system, including utilization of nano carrier delivery to overcome the physicochemical and the biochemical challenges that are there. What are the advantages of the novel drug delivery system? There are multiple advantages that are there, right, starting from the protection from physical and chemical degradation, sustained delivery, improved tissue macrophages distribution, enhanced stability, enhancement of the pharmacological activity, protection from the toxicity, and so on. The therapeutic benefits of the some new drug delivery system is the optimization on the duration of the action of drug, decreased dosage frequency, because we all know that the Ayurvedic, Unani, and other traditional medicines have the higher dosages, the number of frequency of the dosage is very high. But with the novel drug delivery system, we can curtail the frequency of the dosage. Maybe we are developing sustained release, which can have an impact of the drug for a longer period of time, at the site of action where it is required. So decreasing the dose frequency, controlling the site of the release of the drug, maintaining constant drug levels when we are preparing a product for diabetes. So we have to ensure that the constant drug levels at the site of action are there for a longer period of time, so that the effect of the drug is for a longer duration, so that the blood sugar level can be maintained in a sustained manner. The compliance and the patient compliance is again very important, and the safety and efficacy of the product. Again, these all are the therapeutic benefits of the novel drug delivery systems. Recent developments in the novel drug delivery system of the herbal drugs are, I just told you that there are phytosomes, there are liposomes, nanoparticles, nanoemulsions, microspheres, ethosomes, solid lipid nanoparticle, neosomes, chromosomes, and so on. There are metal nanoparticles. There are a lot of advancements. And let me tell you that you are young graduates or postgraduate students. This novel drug delivery, I got to know last night that I have to present on this topic. But I have been following this topic from 2011-12. So when I did my master's in 2011, I was told to choose a topic, and by my guide, Dr. Mujib sir from Jamia Hamdard, he has helped me a lot, and I was very clear about my topic and on my goal that I wanted to prepare a product for acne vulgaris. That was my goal. I just don't want to pass time here when I'm doing my M.Pharm. Many people are coming and doing research which is only in the books or in the papers. So I told that I want to develop a product. That was very clear in my mind. And so sir had given me full freedom. He said, okay, develop, and we will also involve some people from the pharmaceuticals department. So being a pharmacognosis student, along with the pharmaceutic department, at that time I learned how to develop ethosomes. So I developed a product for acne vulgaris, and we had developed ethosomes out of a herbal compound, alpha mangostin. So we isolated alpha mangostin from the fruits of Garcinia mangostana. This is a fruit rind which is found in Kerala and in parts of Thailand and Sri Lanka. This is a fruit, Garcinia mangostana. Garcinia indica is found very popularly in India, but Garcinia mangostana is found only in Kerala. So out of those range, we had seen that alpha mangostin had a very good anti-acne effect on the acnegenic bacteria. And we had compared it with clindamycin, which was the reference standard of the drug which is killing the acnegenic bacteria like Propionibacterium acnes and Staphylococcus aureus. So against these two bacteria, we had tested alpha mangostin, and we found that it has it inhibits the, which is the acnegenic bacteria, up to 98% inhibition. We had reported against the clindamycin, which was around 84%. So at that time, I developed this ethosomes because of the better permeability and solubility into the skin, because we see that the transdermal and the dermal applications, we see that the ethosomes have a major impact. So since then, in the herbal ethosomes, and just not, the research did not end over there. When I completed my M.Pharm thesis and went for the Viva, I had taken the product along with my Viva to show at that time that I have prepared that alpha cure complex also. I registered the trademark, and then I launched anti-acne gel because of the background that I come from, Able Pharmaceuticals. I got the platform. After completing my M.Pharm, I launched this herbal anti-acne formulation into the market with the name of Purodil anti-acne gel, and that contains this ethosomes coming from the alpha mangostin. So from since then, there is a keen interest of in the novel drug delivery system, how it is working, and still I think there is a huge scope, because I have gone through the research papers also. Last night also we were surfing. Since then, 2011-12, and then in 2020, very few research papers are available on the herbal drugs with the novel drug delivery system. There are a few also from Jamia Hamdard or other universities, but I think I still feel there is a lot of scope in it. So I'll come to the types of the novel drug delivery system that are available. Currently, this is phytosomes. Phytosomes are complex phospholipids and natural active phytochemicals bound in their structures obtained by the reaction between phosphatidylcholine and plant extracts in an aprotic solvent. So we can see that here we can see in the diagram flavonoids have been encapsulated, loaded in the chitosome. To form a phytosome, there's a layer which is a hydrophilic layer which is formed. These are the examples. I am quoting with the examples that there are research papers on these that they have been developed. So epigalactocatechin from gallate from Camellia sinensis, we all know, loaded phytosomes which increase the solubility and the bioavailability of the drug. Additionally, showed the physicochemical stability through organoleptic, water content, and physicochemical properties during 6 weeks at various temperatures. Also, we have seen that phytosomes of herbosomes have been developed out of soybean seed, which is glycine max, thermogel, solved the problem of poor absorption and instability and insolubility and fast releasing of the drug. So, also this is another example, rutin from the various citrus fruits that we form have been loaded into a phytosomes for increased solubility. Butia monosperma, जो पलाश पुष्प जिसे हम कहते हैं हिंदी में, gingerol from Gingiber officinale have been loaded into the phytosomes and have showed increased stability, increased bioavailability at the site of action. Then after phytosome, we have liposomes, which are the spherical shaped polar lipid nanoparticles. We can see there is a hydrophilic head and it encapsulates an aqueous core by single or multiple natural or synthetic lipid bilayer. Freely diffuses into its interior, have both hydrophilic and lipophilic groups on the same molecules. So there are different type of liposomes, but we have seen that the paclitaxel from Taxus brevifolia, which is an anti-cancer compound, have been loaded and saturated with the PC-based liposomes have been formed, which is producing homogeneous membrane filtrable drug nanocarrier suitable for intravenous dosing, enhance the solubility, bioavailability, and intracellular uptake and biodistribution. So paclitaxel have been loaded into the liposome form and have been formed to be an intravenous drug dosage have been developed. Then again, different drugs like sterols from have been loaded in the liposomes. Naringenin from immature orange fruits have been developed for enhanced stability and solubility, which also again there are multiple examples which are there for the different drug dosages of liposomes. Then comes solid lipid nanoparticles, which we also called as SLNs, which contains a solid lipid in the center. Solid lipid is a colloidal drug and have been developed in the 1990s. The particle size ranges from the 50 to 1000 nanometer. SLN is processed by using emulsifiers to stabilize the dispersion that is composed of melted solid lipids in the water. Most commonly used methods for preparing SLN is the homogenization. High-speed homogenization is one of them. The application of SLN again, there is a purine from Purida lobata, have been loaded and which have been rapidly and well absorbed, improved the bioavailability more than threefold as compared with that of purine suspension, and increase the tissue concentration in the target organs. So we can see that already people have researched and have been applying this technologies in different aspects of NDDS. Nanostructured lipid carrier, again, we can see here second generation lipid nanoparticle that contains a mixture of solid and liquid lipids, better controlling of the drug release, more stability, enhance the drug loading capacity. And here we use emulsifier along with the solid lipid and the liquid lipid, which we also call as NLCs, nanostructured lipid carrier. The applications of the same is cardamom, we all know elaichi. Cardamom loaded NLC could be used as a food supplement. Thymoquinone from Nigella sativa, which is Kalonji loaded NLC, showed bioavailability and the oral delivery enhancement in the 4T1 bearing BALB mice. So, and improvement in the most liver biomarkers and antioxidant power of thymoquinone was shown. Then, after coming to SLN, there is liposomes. Then comes nano emulsions, which are very common and very a lot of papers have been published on nano emulsions and formation of nano emulsion. Optically single isotropic and thermodynamic stable transparent non-homogeneous colloidal dispersion system. The droplet size is less than 100 nanometers, so that is why because of the reduced and very small particle size that has been more popular these days, composed of the stabilized oil and water, because these are oil and water nanoemulsions, aid of surfactant and co-surfactant and interfacial film molecule is required to form an nanoemulsion. These are all detailed process how we are preparing nanoemulsions and other drugs. And then again, there is one called term called nanocapsules. It is a novel nanovesicular colloidal dispersion system, typical core shell structure. Drug is confined to a reservoir or within a cavity. We can see in the image, surrounded by a polymer membrane or coating. Cavity can contain active substance in a liquid, an oily or an aqueous core, solid form or as a molecular in which the core cell structure is there. Then comes the ethosomes, as I was telling you about that ethosomes, and we use along with the lipids, we use ethanol with it. That is why we call ethosome is a type of liposomes only. It's a novel liposome, a soft, non-invasive lipid-based elastic vesicles developed for topical, transdermal, and synthetic systemic applications. High efficient ability of both hydrophilic and lipophilic drugs. So that is why, because of its ability to compose hydrophilic and lipophilic drugs, ethosomes are much more advanced. Active ingredient delivery to deeper skin layers and the blood circulation. So we have seen that the ethosomes can also reach to the blood circulation. That these studies we have done on the rat skin also, that really whether the ethosomal formulation, the active drug, whether it is reaching to the bloodstream or not. So yes, it is really effective. We can take the drug and we can, in the transdermal application, we can make it reach to the bloodstream also. The examples then there are dendrimers. Dendrimers are also one type of the novel drug delivery system where we have seen curcuma longa have been encapsulated, purarin, silibin, and then liquiratin from glycyrrhiza species have been loaded for different applications for increased solubility, increased bioavailability, and so on. Then there are transferosomes composed of phospholipid and single chain surfactant, provides elasticity and deformability to the vesicles, supplying the nutrients locally to maintain the skin. So again, another form of transferosome where we are using phospholipid along with the single chain surfactant. Then we can see here we are seeing 4 type of novel drug delivery system which are confined in one. One is the conventional liposome, where we see the hydrophobic drug has been encapsulated along with the hydrophilic drug. Ethosomes which contain ethanol in the outer layer and also hydroethanolic solution of the drug is incorporated with the hydrophilic drug. Then we can see single chain surfactant molecule in the form of ethosomes, and also edge activator like single chain surfactant molecule have been used. So we can see there are 4 types in the transferosomes, ethosomes, and the conventional liposomes. What is the difference between all 4 of them? And then neosomes, which are only containing the non-ionic single chain surfactant molecule. Then one of the most interesting ones is the hydrogels, which is also one of the most upcoming. Cross-linked hydrophilic polymer structures imbibe large amounts of water or biological fluids. So formed by a porous 3-dimensional network prepared from cross-linked natural or synthetic hydrophilic polymers, hold large quantities of water or biological fluids in the structure without dissolving. So again, this is a new type of drug delivery system which is gaining popularity. The applications is when it is holding a lot of amount of water in it. So these hydrogel are very much used in the applications for medical dressing, where it is required to hold a large amount of water. Drug delivery in the cardiac repair, neural tissue repair, osteoarthritis, cartilage damage repair. So we can see in the hydrogel also it has been used in the implants like silicone contact lenses, scaffolds, gene and drug delivery, regenerative medicine in the form of hydrocolloids, microfluidics, and also in the contrast, hydrogel containing contrast agents or nanoparticles. So these hydrogel also are gaining popularity. Then comes dendrimers, a micromolecule branch 3D structure, provides a high degree of surface functionality and versatility. Applications for the same are delivery of anti-cancer drugs by dendrimers and dendritic polymers, dendrimers in gene transfection, and we can see that central core along with the encapsulated drug in the terminal groups on the surface. The examples for biopolymer hydrogels are yerbamate loaded calcium alginate hydrogels containing cornstarch, beta carotene loaded rice starch based hydrogel, which increase the beta carotene bioaccessibility that leads to faster digestion of trapped lipid droplets inside starch filled hydrogels in simulated gastrointestinal tract conditions. Then the hydrogel containing crocalinin extract loaded nanoemulsion, cashew gum modified with the phthalate anhydride loaded carboxymethylate cellulose hydrogen, then salic sulfur loaded chitosan based hydrogen. So these are the examples of the hydrogel which have been developed in the different forms. Then comes nanospheres, are also a form of novel drug delivery system which we have, which is the division of polymeric nanoparticle size range from 10 to 200 nanometers, prepared with biodegradable, biocompatible, and synthetic polymers, can be administered orally, locally, and systemically. Release the encapsulated drug in a slow and sustained manner, and it has been used and can be used in the target organs like liver, spleen, lungs, and the spinal cord. We can see that the difference between the nanosphere and the nanocapsule, where the in the nanosphere the drug has been encapsulated with a polymetric matrix, and whereas in the nanocapsule there is a polymeric membrane. Then also one of the most interesting, which is the metal nanoparticles, very much a very traditional system of medicine. Although we call it a novel and the more advanced system of drug delivery, but this metal nanoparticle, I must say, that has been referred in the Ayurveda system since ages in the form of Swarnbasam that we have been using since ages and has been recommended. And these are nothing but a novel drug delivery system, a nanoparticles that has been formed in the Ayush system since ages. Like silver nanoparticles have been developed, gold nanoparticles have been developed in as the form of Swarnbasam since a long time. Copper nanoparticles, zinc oxide, which is the Yashad Basam that we have been using. Then there is iron oxide nanoparticles, that is Loha Basam that have been using. These all metal nanoparticles have been mentioned in the ancient text 5,000 years ago, and they were nothing but nano formulations only. So we call it novel, but still we are only researching them out, because we are researching them, because they are already mentioned, their use have been mentioned, and they have been used in the different applications and different diseases at different point in time. Titanium dioxide nanoparticles, and also we can see the structure here is for the gold nanoparticles. Also, I'd like to share one development which have been in the applications. They can be utilized for achieving the intracellular drug delivery and controlled release through a photothermal route, metal nanoparticle integrated multifunctional liposomes to improve intracellular drug delivery, overwhelm the multi-drug resistant. As we can see that the drug resistance MDR is again increasing day by day, and we have seen that the metal nanoparticles have a big role in the MDR, be it tuberculosis, be it antibiotic resistance for the infectious diseases. So applications for these nanoparticles are having immense usage in the therapeutic segment. Then again, Amaranthus caudatus pod extract loaded silver nanoparticles, brown seaweed loaded zinc oxide nanoparticles, curcumin loaded iron oxide gold core cell nanoparticles. So multiple nanoparticles are in the phase of development, and once one application that we also have developed, I will share with you that gold nanoparticles currently we have developed at ML Pharmaceuticals, and we have incorporated into one of the products, which is our sparkling gold face wash, which is you are using under the trade name of Ayurveda. And we have developed the Swarnbasam into the form of the nanoparticles, and that Swarnbasam has been included. And we have seen a lot of research papers which is recommending that the Swarnbasam or the gold nanoparticles are having very good therapeutic activity in reversing aging. Aging ko reverse karta hai. And that is why we have incorporated this gold nanoparticles in the form of a face wash and a day cream and a night cream. So that was done by us. So these are the two developments that we have actually applied in the market, not just talking in the air or in the PPTs. So we have actually applied and we are actually making it available in the market. So again, one more category, which is biopolymers, are natural polymers produced by the cells of the living organisms, consist of the monomeric units that are covalently bonds in chain to form larger molecules. Main classes of biopolymers classified according to the monomers used and the structure of the biopolymers formed. I'll just share the examples of the beta carotene, yerba mate, the hydrogels, cashew gum hydrogel, carboxymethyl cellulose hydrogel. The common biopolymers are which have been used are the collagen, silk fibrin, and gelatin. Collagen is the primary structure of the vertebrates and is the most abundant protein in mammals. Silk fibrin is another protein-rich biopolymer that can be obtained from different silkworm species. And the applications for the same are the biomedical due to their biocompatible properties. Biopolymers are vastly for tissue engineering, medical devices, and pharmaceutical industries. Collagen-based drug delivery system and chitosan as an antimicrobial agent have been used. We can see here in the picture that the different biopolymers we are isolating from, which are plants, cellulose, starch, gum arabic, algae, which is agar, alginate and carrageenan. Animals, we have isolating the chitin, collagen, and hyaluronic acid, which is much more very good for the skin and very popular these days. Fungi, we are isolating chitosan, and then bacteria, gelan, xanthan gum, and dextran we have been using since a long time. So these are some articles which were recently published in different media, which are the gold nanoparticles hold promise in the combating aging. In the European researchers have told that gold nanoparticles can help arrest aging, and also in the different papers and their research has mentioned that these gold nanoparticles have a very good effect on the skin. Also, one of the professors, Anand Chaudhary sir from BHU, he has also done and published a paper on the assessment of antioxidant and the dermoprotective activities of gold nanoparticles as safe cosmetic ingredient. So not only here us at the BHU also is researching on the same terms, and we have seen that the gold nanoparticles are having a use in the market already. Also, when we are talking about the novel drug delivery system, artificial intelligence cannot be left behind because this AI is now more and more in use. Although you must be using AI for your current assignments or maybe in the practical life these days. But yes, AI is also, and the machine learning have been used in the labs now, and artificial intelligence and machine learning, they are increasingly used in the pharmaceutical research, development, drug discovery, even in clinical trials and data analysis. These technologies can efficiently process vast amounts of data, identifying patterns, predict outcomes, and we used to do the in vitro studies, but these days, along with the in vitro studies, we have used to use a lot of kits, biochemical markers, but along with the artificial intelligence, we can see, we can estimate the enzymatic activity, whether it will be binding with the protein or not, the protein binding activity. We can see a lot of activities, in vitro activities, which we can do with AI these days. AI in drug discovery, we can see that AI in drug design, predicting the 3D structure of the target protein, predicting the drug protein interactions, AI in determining the drug activity, AI in the de novo drug design, and also in AI in polypharmacology, in chemical synthesis, in drug repurposing, and drug screening. Everywhere there are multiple applications where we can use AI to develop these novel drug delivery system and also to formulate the therapeutic effects of the same. So conclusion is that as we know that the herbal pharma industry is growing at the CAGR of 17%, and herbal market is going to be big, very big in the times to come. And with the ancient system of medicine, which is the Ayush system of medicine, I feel there's a lot of scope where we can take the medicines and follow the proper research protocol, as Dr. Pandey has already told you about how the development of leucoskin he has done, right from the in vitro studies, in vivo studies, clinical trials, right from the scratch. He has done the studies, And that is how I think we need to develop our herbal products, which are scientific, research driven, and driven by the novel drug delivery system, and also the use of artificial intelligence in the same. So developing herbal drugs and according to the market and the need of the market is the need of the hour. And we can see that in the export also there is a huge scope of the herbal drugs. We are currently exporting around $1,250 million products which are being exported by of the Ayush industry in the globally in the different countries. But that is just only 3 to 4% of the market share. The remaining market share is with the China market. Chinese herbal traditional medicine are doing much better than us, and I feel there's a huge scope there also. So the globally also, in India also, the market is big. We need that we all, whether you are an undergraduate or a postgraduate or a scientist or a professor, together I think we can bring about a change how people take about these herbal drugs, and we can develop some novel drugs in terms of therapeutics, in terms of their drug delivery, in terms of their dosage forms, in terms of their frequency of dosage. We can make it more aligned with the modern system of medicine, and we can also develop those protocols which are like sir has told about the clinical trials. We are also now going about those, following the parameters of the CTRI and the other regulations. So following all, I think we will be at par with the modern drug system very soon. Thank you very much. Any queries? Any queries related to?
Yeah.
Can you come again?
Are you using loading the nanoparticles onto the hydrogen?
Hydrogel?
Yeah.
No, hydrogel is a totally different concept where we are loading the excess amount of waters into the lipid layer. And here gold nanoparticles is a different concept where we are developing Swan Basam. There's a method where we develop the Swan Basam, encapsulate the drug into the emulsions, into the different emulsions, that in the condition of face wash where it is we are using different surfactants. We absorb the Swan Basam into that surfactants and then use them in the product. So hydrogel is different. Nanoparticles of gold nanoparticles are different. So they are all both different. Any other questions?
Yeah.
Thank you, sir, for this very informative session. Now, I would like to request Madam Swati Madan and Dr. Vikesh Kumar Shukla, sir, to felicitate Dr. Sanchit Sharma. I would like to call upon Nirupma ma'am on the stage. Can we have a big round of applause for them? Thank you, sir. Simultaneously, by today afternoon itself, track 1 oral and poster presentation was held, which was coordinated by our faculty coordinator, Dr. Kavita Munjal, where total 28 presentations were successfully evaluated by our respective evaluators. To felicitate them, I would like to request Dr. Tanveer Navis sir and Dr. Vinay Lathar sir to kindly approach to the stage to felicitate Dr. Shaukat Rasoor Mir and Dr. Maqsood Ali. Can we have a big round of applause for them?
Thank you very much for all. Thank you. Sponsor है डॉक्टर संचित भी यहाँ मौजूद हैं. Audience, this जो अभी जिक्र हो रहा था AYUS Veda जो डॉक्टर संचित कर रहे हैं, and that gift from the Dr. Sanchit company AYUS Veda Able Pharmaceutical. So big round for applause for the Dr. Shaukat, may I request you?
A big round of applause for Dr. Shaukat Rasool.
My personal pleasure because Shaukat and I are the batchmate of B Farm and 1995. 95 to 99 and then 99 to 2001 M-Farm Pharma in Jamia. Thank you. Thank you for volunteer bill. Thank you. क्या बताता हूँ
Thank you, sir. Moving ahead, I would like to request Madam Mariam Sarwat to introduce our next speaker, Madam Dr. Ghazla Javed.
Thank you, audience, for listening the eminent lectures which has happened just now. After the three marvelous lectures by our eminent speakers, Dr. Hemant, Dr. Suhail, and Dr. Sanchit, now we are heading towards another fascinating speaker, Dr. Ghazala Javed ma'am. What a coincidence! She is also an alumnus of Jamia Hamdard. Apart from that, she has always been my great mentor when it comes to research in Unani medicine. So we have a brief introduction about Dr. Ghazala Javed, although there is a lot to say about her. Dr. Ghazala Javed is working as Assistant Director, Unani, in the headquarters of Central Council for Research in Unani Medicine, Ministry of Ayush, Government of India, where she is looking after technical work and is involved in planning, implementation, coordination, and monitoring of research and development, fundamental research, drug standardization, quality control, and other related activities of peripheral institutes. She is also in charge of Drug Standardization Research Unit, New Delhi. As a nodal officer, she is coordinating activities of premier institutes of CCRUM, the National Research Institute of Unani Medicine for Skin Disorders, Hyderabad, and Regional Research Institute of Unani Medicine, Mumbai, and JJ Hospital, Mumbai too. She is also a Unani specialist in the clinic in Goa. During her 27 years of career, she has participated in many professional meetings and conferences and made a niche for herself as an expert from CCRUM and Ministry of Ayush. She has bagged a gold medal in her academic career at Jamia Hamdard. She holds a PG diploma in intellectual property rights and WHO fellowship in managing public health programs. She's a trained bioethicist also from NIH USA. She has published around 90 papers in reputed international and national journals, participated in more than 200 national and international scientific platforms, and edited around 20 books. During her tenure of 8 years in the Ministry of Ayush, Government of India, she looked after the work related to international cooperation and actively contributed to the globalization of Ayush systems. Amongst other areas, she dealt explicitly with work related to American division, UN bodies like WHO, WIPO, UNESCO, etc. She has participated as delegate from India several times in the intergovernmental committee meetings as protection of traditional knowledge, traditional cultural expressions, and folklore in WIPO, Geneva. She has also involved in developing national position papers on protection of traditional health knowledge with a team from GNU. She has also served as an advisory member for the Forum of Indian Traditional Medicine, FITM, for the Ministry of Ayush. And this is not enough. There is further. She has been a scientist to the National Center for Natural Product Research, University of Mississippi, Oxford, USA, where she had active interactions with US FDA. Also worked as a consultant for Ministry of Ayush for assisting the Indian Institute of Foreign Trade, IIFT, for export potential for Ayush products in SAARC and ASEAN countries. She has the honor of featuring as the face of Unani in the book India's Most Powerful Women, written by Ms. Ahluwalia, where she has been featured along with eminent personalities like Sushma Swaraj, Kiran Bedi, and Menka Gandhi as well, amongst many other eminent personalities. The book was released by Secretary General of United Nations, Berlin, Germany, and was also presented to the then Honorable President of India, Shri Pranab Mukherjee, at the Rashtrapati Bhavan. She was a special invitee to interact with President Obama and Michelle Obama during their visit to India in January 2015. She has been conferred Make in India Award for brilliant contribution in the field of Unani medicine by Honorable Minister Shri Ashwini Chaubey and the NCR Delhi Ratan Award too. She has also been awarded one of the most prestigious Dr. Sarojini Naidu International Award for Working Women, which was presented to 100 most important women from 15 countries of the world by Asian Academy of Arts at the International Chamber of Media Industry. So I invite you, ma'am, to give your enlightened lecture on the topic Recent Advances in Unani Medicine.
Thank you, Dr. Mariam. Hope I am audible.
Yes, ma'am, you are audible.
I'll just share my screen. Just confirm that. Can you see my screen?
Yes, ma'am.
Ma'am, the screen is visible and you are audible. You can start the lecture. मैम को एडमिट करूँ शेयर तो है ना मैं फोन करती हूँ कहाँ से क्या कह रहे हैं आपको अनम्यूट करना है हाँ आवाज नहीं आ रही first. Pause are you speaking to me? Ma'am have network issue. हाँ ma'am बिल्कुल आपको mute paused. Ma'am is disconnected. अच्छा मैम कोई नेटवर्क इशू है क्या Students, please have patience. All the delegates, please have patience. Please be seated. The lecture will be starting in any moment.
Am I audible now?
Yes, ma'am, you are audible.
I've tried to join with my phone. I don't know what's the problem. 1 second. I'm sorry for this glitch. My slides are visible?
Yes, ma'am, slides are visible.
Okay. So, kindly let me know if again there's some problem.
Okay, ma'am.
I don't know from where I was audible and where. So, to begin again, significance of traditional medicine, I was talking about it. And so, as I was mentioning, that this has been recognized at UN level. And in the declaration of Astana, it was very clearly mentioned that success of primary healthcare can be driven by applying scientific as well as traditional knowledge. And this very much goes with the thread of today's session, which is traditional science knowledge and ancient wisdom. So around 88% member states have acknowledged their use of TNCM, which correspond to around 170 member states. And we are also aware of this WHO traditional medicine strategy, which also envisages about integrating TNCM into national health systems, and that WHO will facilitate national policies in that regard. And then mainstreaming of Ayush system in public healthcare is one of the key strategies of Government of India to provide healthcare to masses, for which ample policy support is provided. So, looking at Ayush in India, we heard our earlier eminent speaker talking on various initiatives which have been taken. So, this Ayush has been elevated. Earlier, it was a department as a full-fledged ministry in the year 2014, and since then, many milestones have been achieved. And as we might have heard, very often, Honorable PM have always given attention to the importance of evidence-based research and innovation. Maybe I'll just switch off my video so that, yeah. Yes. So, recognizing this global shift towards chronic and non-communicable diseases, Ministry of Ayush emphasizes integrating traditional systems with conventional medicine to promote a holistic and patient-centric healthcare approach. We are getting ample social and political push, as you can see from the different quotations of Honorable PM of India and Honorable Union Minister of Ayush. To quote some, sector of traditional medicine is expanding continuously, and we have to take full advantage of its ever possibility. And India's traditional medicine system is holistic science of life. And by giving the slogan of Our Planet, Our Health, WHO has promoted India's vision of One Earth and One Health. So similarly, as to those to whom Unani medicine is new, just to give you a brief sensitization on this, science of it is science of which we learn the various states of body in health and when not in health, and the means by which the health is likely to be lost and when lost is likely to be restored. So it's flourishing in India since 8th century, and we can very well say that with the ample government support and the strong infrastructure, India is world leader in Unani system of medicine today. These are the basic concepts and principles where we believe in preservation and promotion of health, prevention of diseases. This is the diagnosing methodology. I won't go in detail in this. These are the four treatment modalities which we adopt: dietotherapy, pharmacotherapy, regimenal therapy, and surgery, which is being conducted to for as preventive measure as well as for providing therapeutic modalities. So basically, in a nutshell, we can say that Unani approach is toward preservation of health, prevention of diseases, and restoration of healthy lifestyle. And preventive, promotive, it provides preventive, promotive, curative, and rehabilitative healthcare with holistic approach, which is very much required when we are talking about mainstreaming. Here we believe in strengthening the Tabitte Mudabbira Badan, which we call as a natural power in your body to fight the disease and maintain health. So, in Unani medicine, if we see at national level, today it is an integral part of national healthcare delivery system. The organization from which I am coming strives for excellence and global leadership in the field of Unani medicine by comprehensive research for quality assured and cost effective products and adopting modern integrative approach in clinical practice, research, education, and public health programs. This is our organization from which I am coming. Our presence is almost pan India. We have a strong network of 23 peripheral institutes, out of which one is at national level Hyderabad, central in Lucknow, 9 regional level, and one regional RRC in Allahabad, and 7 CRUs. We also have drug standardization research unit and institutes. These are the pictures of some of our institutes on CCRM at Hyderabad, Aligarh, Bhadrak, Chennai, just to give you an idea what kind of infrastructure we have available in Unani medicine. Again, in Lucknow, Mumbai, Patna. So we are engaged in these basic, apart from this, we also carry out many other activities, but these are the basic research programs in which we are engaged, which preclinical and clinical pharmacological research, fundamental research, drug standardization research program, survey and cultivation of medicinal plants, literary research program, IC activities, and public health program. Our peripheral institutes are also engaged in multifaceted activities encompassing all these programs and other activities also, which is mandated to them from time to time. This is just a snapshot of some of the activities. So, modes of research which are being carried out at CCRM. So, projects as per program and annual action plan of CCRM are allotted. So, we have one IMR policy where we have IMR projects which are developed by the peripheral institutes itself, collaborative research projects. We have expression of interest scheme for which we advertise on our website and call for applications under this scheme. And then also EMR projects under IURGAN scheme, which was earlier called as EMR scheme. So under preclinical studies, what we are basically doing toxicological and pharmacological studies at our 2 centers, one at Hyderabad and one at Srinagar. These are the laboratories. Just to give you an idea, this is our institute in Srinagar. This one in Hyderabad. You can see our scientists working. Then these are the clinical research program. One of our mandate is to document safety and efficacy of pharmacopoeia classical Unani drugs. As you are aware, these Ayush systems have heritage of classical texts, whether it is Ayurveda or Unani, and under that, those formulations are used since many years. So we are documenting that and also working towards of safety and efficacy, and also working toward new drug development for emerging diseases and diseases of national importance. So it was a conscious thought that we should utilize the full potentials of our researchers who are working in our peripheral institutes. So we have given them the opportunity to think out of box and develop IMR projects. They can have a co-PI from other institute and they can submit this IMR project, which is considered, and the policy aims to conduct studies in a time-bound manner on innovative ideas. Then in collaborative research, I think our earlier eminent speaker, Dr. Suhail Parvez has mentioned, because there was net problem, but I could hear that he was telling something about the collaborative projects only. That is one of the example, because we have given many collaborative projects in Jamia Hamdard also, MAT also. So basically, the objective is to establish firm links with state-of-art institutions of excellence, which have expertise and facilities in order that collaboration research provides globally acceptable results, and to generate adequate statistical data, and to make scientific exploration of IU system. So this scheme is available in our website. You can go and visit and can apply also. Again, this EMR scheme which I was mentioning, this is also basically to support education, research and innovation in Ayush. Under this also you can submit EMR projects for Unani stream. CCRM is managing it on behalf of Ayush, Ministry of Ayush. So the recent research trend which we are following is new drug development for emerging diseases like anti-dengue and anti-cancer drugs, and evaluation of efficacy of Unani drugs in neurodegenerative diseases like dementia, Alzheimer's, and redesigning of dosage forms of the Unani drugs, especially for diabetic population. And finding, because as you might be aware, Unani drugs are mostly sugar based. So it is really very important that we redesign these dosage forms, especially for diabetic population, and we are working toward that, and finding prophylactic solutions for lifestyle disorders, immunomodulatory role of Unani drugs, shelf life studies of various Unani formulations, and we are also involved in public health related activities. The significant leads achieved in these are the names of some of the diseases which we have, where we have obtained some leads, and 18 patents have been granted to CCRUM, and 10 patent applications have got approval from National Biological Authority of India. Another area which is of importance is fundamental research program, where we are working towards developing a field of the Unani concepts as Unani genomics, and you might have heard about IU genomics. On the same pattern, we are working because we also have this concept of ACLAR, and where we study the clinical, physiological, pathological, biochemical, and genetic parameters of the subjects of different temperaments, and conducting clinical assessments of Mizaj and genetic studies on the theory of humors with special reference to diabetes mellitus, hypertension, vitiligo, and psoriasis. So recently we have also signed an MOU with CCMB Hyderabad, which is one of the institute engaged in conducting molecular and genetic studies. So we want to expand under this area, and we want to establish a center of excellence for fundamental research program, where we'll be conducting many omics studies, whether it is proteomics or metabolomics or genomics. So we are working toward that. Then drug standardization, of course, we are developing standards, pharmacopoeia standards, and national formula of Unani medicines, essential drug list. So we have a dedicated lab in Ghaziabad and one at headquarters only. And so we are following all the recent and trends by PCIMNH, which is the apex organization in Ghaziabad, and we are conducting all these tests and developing the pharmacopoeias. So when we are talking about integration of Unani medicine in health services, you can see that there we have a physical integration of facilities and health services, and we have functional integration for achieving national health goals. I will be elaborating a little bit in my further slides. NPCDCS program, education integration by inclusion of modern medicine components in Unani course curricula, clinical practice integration through co-location centers in the tertiary care allopathic hospitals, and research integration through interdisciplinary and collaborative research with integrated protocols, and standard setting integration for quality assurance in products by using modern technical tools. So this is the example of a National Program for Prevention and Control of Cancer, Diabetes, Cardiovascular Disease and Stroke Program, where this was for the comprehensive with a holistic approach management of lifestyle related disorders, and 6 districts were adopted, where CCRM has adopted 1 district through its Lucknow Institute at Lakhimpur Kheri. The main purpose was to synergy be to harness between the allopathy system under NPCDS program and Ayush. So we were offering the prophylactic measures offered by each Ayush system along with yoga program, and government is working toward that. We got some encouraging result in this. This is the example of integration in clinical practice which I was mentioning. We are present in the prominent allopathic hospitals, central government hospitals, Dr. Ram Manohar Lohia Hospital, Deen Dayal Hospital, Upadhyay Hospital, Safdarjung Hospital. These units are managed by, centers are managed by the council, and we do get referral patients also from, our allopathic counterpart, and we are managing those patients. So these are the good examples, and we are getting some encouraging result in this integration in clinical practice. Also, when we see about integration in collaborative research, so I had just mentioned, basically we are establishing strong linkages with other premium institutes. So we are inviting the projects, we are evaluating it through multidisciplinary expert committee, then we are signing the MOU, allotting the projects. So scientists are working close connection with headquarters and peripheral institutes. Final outcomes of the projects are published in the form of research papers in the journals of international and national repute. And here I would mention that our council also bring out two journals which are UGC care listed. One is in English language and other one is in Urdu language. The English one is Hippocratic Journal of Unani Medicine. The results are also disseminated through various platforms. These are mention of some of the institute where we have collaborated. We have signed many MOUs with MIT, Jamia Hamdard also. These are some of the drug testing laboratory at RRI in Chennai. We are in the process of getting it upgraded to next level because this institute has come up with getting, this was the first institute to get NABH accreditation and NABL accreditation, and it will be also developed very soon as Center of Excellence for Regimen Therapy. Ghaziabad lab. Then this is also are important. We all know that medicinal plants are the backbone of traditional medicine. So our premium institutes are engaged in conducting survey and cultivation for sustainable conservation and development of medicinal plants. We are maintaining the data by way of repository. These are some of our publications. And we are bringing standards into healthcare system, and all our institutes are in process of getting NABH and NABL accreditation. So you can see here, and rest of them also will be getting accredited very soon. So we are also integrated in the digital health landscape also. So there is one important project of Ministry of Ayush, which is called as Ayush Grid project. Under that, we have taken many IT and AI-based initiatives. To begin with, we have implemented Ayush Hospital Management Information System in all our peripheral institutes. And the Unani mobility codes are uploaded in National Ayush Mobility and Standardized Terminology electronic portal. We have released certain ebooks and apps. This is one of the ebooks, and it is an example of intra-Ayush collaboration. This where Ayurveda Council has worked on this book. There is one Unani doctor working over there, and CCRUM has launched this book. This is one of the most important book of Unani medicine. This is available in public domain. Again, this is also developed in this Unani cell of Ayurveda Council and released by CCRUM. These are the apps which are freely downloadable from Android and iOS. So there is one application web-based, Know Your Temperament, where you can click on this link of CCRUM and you can know your temperament by taking part in interactive questionnaire. Then you'll come to know what is your temperament and what all you should take and not take, advice is given. And rest of them, this common remedies in Unani is for general public, where easy to use ingredients are mentioned for common ailments which can be used. Unani treatment guidelines and single Unani drugs and Unani Murakkabat or formulations are very handy for all Unani stakeholders. This is at your fingertips. You can just download it and see what all is prescribed in Unani system of medicine. And Unani Murakkabat also, we have mentioned the ingredients and all, and it's quite handy. And we look forward to have a good feedback from you, because we are very soon planning to bring revised version also. So I request all of you to download it from iOS and Android. These are freely downloadable and make use of it. Then these are some of our publications. So we are also venturing into education through our two institutes, one at Hyderabad and Srinagar, and offering MD courses in Unani medicine and Unani pharmacology, Ilmul Adviya, and normal pharmacology also. So if they are affiliated to these two universities and conducting these courses. We are engaged in many IC related activities. Ayush Grid, as I was mentioning again, elaborating on that, we are here very much, and Ayush Next portal also, where we are giving, it's an informative exchange, giving career advice, what all scope is there in all Ayush system, what you can do there, and we are spreading awareness in form of quizzes and blogs. So you can visit this. So we have also almost completed the systemized nomenclature of medicine clinical terms project, and this has been very much appreciated. This was completed in time, and we have total of Unani terms have been translated and incorporated in Ayush extension of Snowmed City. Same with Ayush Bharat Digital Mission, our healthcare facilities have been registered in this so that you can know where are these facilities available, how many doctors are there, whom to approach, and how to approach. So at international level also, if we see the recent survey done by WHO mentions that use of Unani medicine has been reported by 82 member states, and in the traditional medicine chapter under ICD-11, it is a formative step, which is international classification of diseases, to provide the means for doing research and evaluation to establish its efficacy, to know the usage of a particular Unani morbidity code, how many diseases are being, you are treating or visiting a particular healthcare facility. So that has been launched. In detail, I'll be telling about these WHO benchmarks also here about this ICD-11. So this inclusion basically acknowledges the richness of traditional medicine and foster collaboration advancing global health standards. So this was very much appreciated and a very laudable work of Ministry of Ayush. These are the benchmarks for training of Unani medicine. If somebody wants to plan training at national or at any of the WHO member state, they can plan the training and deliver it to a plan and advice according to the number of hours, and competency based knowledge and skills is provided and content structures. Same here with practice of Unani medicine. Levels of practices are mentioned, infrastructure requirements, what all are required, even for regimen therapy, cupping. You can download this and you can see, it is very handy for Unani stakeholders, regulators, and policy makers. Then WHO has also released international standard terminologies on Unani medicine. So this will facilitate Unani medicine professionals, policy makers, health workers, service providers, researchers, and the general public to use the same concept, understanding, to bring out the uniformity. So I request everyone to use these terminologies when they are publishing any kind of paper on Unani medicine. These terminologies are there for Ayurveda also, so you should refer to them. That will be in your research paper along with the book. So we have also entered into dialogue with the Ministry of Health and Medical Education in Iran, signed an MOU with Tajikistan, set up a Unani academic chair in Bangladesh, and earlier there was a chair in University of Western Cape, South Africa, where Unani medicine is recognized, and our undergraduates can go and practice over there. So, in a nutshell, we all know that traditional scientific knowledge and ancient wisdom can play a pivotal role in promoting health and wellness and achieve the Sustainable Development Goal 3 for good health and well-being. And our traditional medicine emphasizes healthy lifestyles for disease prevention and lifestyle modifications. And the global medical indicators show a shift from medical care to healthcare, encompassing preventive, promotive, and rehabilitative aspects, which are the mantra of these traditional medicine. And the modern approach based on the stem cell or traditional system medicine through proper integration of this solution will provide solution to challenging health problems. So indeed, there's a worldwide trend towards utilization of traditional medicine. And as our earlier speaker was also mentioning, that there's a need for scientific validation of safety, efficacy of traditional formulation techniques, and we are doing it along with our in a collaborative mode with our premium academic and scientific organization. And India has well developed infrastructure for academic research and healthcare institutions. And CCRUM is making efforts to find solutions through scientific collaboration. And I would say the government of India's efforts towards strengthening of Unani educational system and multifaceted development has made Unani medicine an integral part of national healthcare delivery system. Thanks. I would have loved to be present physically there, but I hope I was able to do some justice. I'll just stop sharing my screen from here. There was very network glitches happening. I have to shift to boardroom of this hotel where I've just checked in.
Thank you, Ghazala ma'am, for your very informative lecture about the various schemes of CCRUM as well as Ministry of Ayush. And especially the digital health landscape and apps developed by CCRUM. I'm sure the delegates will be greatly benefited by the information provided by you. And ma'am, although you are traveling to Bangalore, but you have taken time for our conference, it shows your commitment to research and your close association with our Deputy Dean, Dr. Tanveer Naved, sir.
Thank you so much. So next time I look forward to be present physically amongst you.
Yeah, ma'am. We also want you to be here so that we can interact with you physically
Yeah,
and
sure.
all the people of our department can meet.
Yeah. Thank you for providing me the opportunity. Thanks to you. Thanks to Dr.
Most welcome, ma'am.
Naved. Thanks to everyone, organizing secretariat, everybody.
Thank you, madam. Now I request Dr. Vinay Latha sir to felicitate Dr. Ghazla Javed ma'am. And I also request Dr. Tanveer Nave sir to receive the felicitation on behalf of madam. And Dr. Chitme sir. And
I must thank Dr. Naved for
Dr.
that.
Vikesh Kumar Shukla sir.
Thank you very much, Rala ma'am, for this wonderful talk. And we are, all the people hearing very, with very patience here. And we are very delighted to know all the recent advances in the Unani medicine. So thank you very much. And I'll be receiving on your behalf and ensuring that I'll come to you to bring all the things. Thank you very much.
Thank you so much. So kind of you, So kind of you. Thanks.
Can we have a big round of applause for madam?
Thank you. Thank you so much. Once again, thanks to Professor Tanveer Naved.
Birthday wish sent to you. I'm sure you'll receive it in Hamdard. सभी ले लीजिए कर लेंगे
सर आप हमारे moving ahead to felicitate our session chairs, I would like to request Dr. Tanveer Nave sir, Dr. Chitme sir, Dr. Vinay Latha sir to felicitate Madam Dr. Harsha Khadwal. मैम सर एक एक करके नहीं नाम मैम का अरे मैडम का Can we have a big round of applause for Madam Harsha Khadwal, a session chair?
फोटो फोटो एक फोटो ले लेना एक फोटो अभी इमीडिएटली देना बता
I also request her to please felicitate Dr. Mariam Sarwat, our session chair. Madam Dr. Mariam Sarwat. Can we have a big round of applause for Madam Mariam Sarwat? Thank you, sir. By this, we came to an end of this particular session, track 1. And we have arranged a high tea for all our participants. You may please approach to the E2 auditorium hall for the high tea. Thank you.